Hei og hjertelig velkommen til åpningen av det nye KG Jebsen-senteret for Alzheimers sykdom. Jeg heter Emilie Ranheim-Skyten. Jeg er lege, men også en av hjerneforskerne ansatt på det nye senteret. Og det er derfor med ekstra stor glede at jeg i dag skal stå her som deres konferansier og geleide dere gjennom dagens program. På programmet i dag så står det blant annet en høytidelig båndknytingssermoni. Forskerne på Jebsen-senteret skal få lov til å fortelle dere litt om hvordan de jobber for å komme sammen om å løse den store alzheimergåten. Men kanskje helt spesielt er det at i dag får vi besøk av verdensledende forskere innad i sitt fagfelt, som skal fortelle oss litt om hvordan det står til med Alzheimers sykdom helt fra forskningsfronten. Og i tillegg til det, så er det sånn at dere som deltager i dag har muligheten til å direkte interagere med disse forskerne gjennom appen Slido. Og tilgang til Slido, det finner dere i linken i YouTube-kanalen her, eller dere kan vende mobilkameraene deres mot QR-koden, som dere også finner på programmet på nett. Helt først så vil jeg bare si en kort beskjed på engelsk. To all the English-speaking viewers we have with us today, I just want to inform you that even though the initial part of this opening ceremony will be held in Norwegian, the scientific lectures will be in English and subtitled in Norwegian. So please don't hesitate to stay for the lectures, and please send your questions via Slido in English, if you please. Helt først på programmet vårt så skal vi høre noen ord fra professor og Nobelprisvinner Edvard Moser, som også er den nye lederen for senteret vårt. Vær så god, Edvard.
Enda. Men det vi tror er at vi kan gjøre noe, og vi har noen knagger å henge ting på. Så et av utgangspunktene som vi tror er viktig her, det er at Alzheimers sykdom, det er en sykdom som begynner i et spesielt hjerneområde. Nesten alltid, ikke alltid, men nesten alltid. Og det hjerneområdet, det er hippocampusregionen og entorhinal cortex-regionen som er i tinninglappen og langt bak i hjernen vår. Det området er tilfeldigvis akkurat det samme området som vi på Kavli Institute har forsket på i snart 20 år. Og der vi fant at området og celler i det området er viktige for å lage stedsans, for å lage hukommelse og for å gi oss en fornemmelse av tid. Så vet vi også at akkurat det med sted, hukommelse og tid er også det som blir rammet i Alzheimers sykdom, så det er tydeligvis en kobling der. Det vi nå ønsker å gjøre i det nye KG Jebsen-senteret, det er å koble den forskningen vår, den grunnforskningen vår, til klinisk forskning på Alzheimer. Det er en såpass opplagt link der at vi kan ikke la være å forfølge det. Så vi ønsker å bringe den forskningen vår på Kavli Institute til klinikken, knytte de to feltene sammen, slik at vi kan bidra til å forstå hva er det som går gale, og kanskje særlig i akkurat de to hjerneområdene som jeg nevnte. Og så at man derigjennom kan komme på spor av mekanismene, og til slutt da forstå hva som går galt, og finne en kur helt til slutt. Naturligvis så skjer ikke alt det her på Kavli Institute, eller på de som vi samarbeider med, men vi håper å kunne bidra vesentlig. Og for å få til det her, så må Kavli Institute samarbeide, og det vi gjør i Jebsen-senteret, er at den forskningen vi gjør på rotte og mus, den skal vi også gjøre hos mennesker. Vi har da i samarbeid mellom NTNU og St. Olav og Helse Midt-Norge, så har vi fått en 7 Tesla MR-skanner med veldig høy oppløsning, sånn at vi kan skanne menneskehjernen mens folk gjør forskjellige typer oppgaver. Og vi kan skanne både normale folk og folk som har sykdommer av forskjellige slag, inkludert tidlig fase av Alzheimer, eller folk som er i risikogrupper for Alzheimer. Og ved å knytte sammen alle de nivåene, fra mus og rotte, til den normale menneskehjernen, til den den syke menneskehjernen, så tror vi at vi kan faktisk begynne å linke nivåer sammen og forstå mekanismene som ligger til grunn for patologi i menneskehjernen som leder til Alzheimer. Vanligvis når vi åpner en bygning, eller når vi åpner en bru, eller hva det skal være, så er det vanlig at man klipper av en snor. I vårt tilfelle så er det litt annerledes. For her, i det nye Jebsen-senteret for Alzheimers sykdom, så ønsker vi å knytte sammen flere miljø som tradisjonelt ikke har arbeidet sammen. Det er grunnforskningsmiljøer ved Kavli Institute, som har jobbet med hjernen sine mekanismer for sted og for hukommelse og for tid. Og det er de kliniske miljøer som tar imot Alzheimer-pasientene, prøver å finne ut hva som går galt, og om man på sikt kan utvikle en kur. Så for å symbolisere det, så vil vi i stedet for å klippe et bånd, så vil vi knytte sammen bånd for å illustrere det å bringe sammen kliniske miljø og grunnforskningsmiljø. Så derfor så har vi fått rektor Anne Borg fra NTNU og administrerende direktør Grete Åsved fra St. Olavs hospital til å komme sammen og knytte sammen et bånd som skal da symbolisere starten på det nye Jebsen-senteret.
Først og fremst må jeg si gratulerer med dagen. Dette gjør meg stolt som rektor. Jeg synes det er fantastisk å se hvordan våre fagmiljøer vokser og styrker seg og samarbeider om å skape fremragende forskning. Så er det jo ekstra flott denne gangen å knytte et bånd heller enn å klippe en snor. Dette synes jeg er en viktig gest som demonstrerer nok et godt samarbeid mellom St. Olavs hospital og NTNU. Vi har en stor fordel som universitet og forskningsinstitusjon å ha et tett og godt samarbeid med St. Olav. Jebsen-senteret for Alzheimers sykdom er et nasjonalt, tverrfaglig forskningssenter som forener forskere i det felles målet å forstå utbruddet av Alzheimers sykdom og de tidlige stadiene av sykdomsforløpet. Og jeg må si at jeg imponeres over visjonen til dette Jebsen-senteret, nemlig at den skal kombinere nobelprisvinnende grunnforskning fra laboratoriet til pasient. For å oppnå dette samarbeider senteret med eksperter i flere prosjekter for å bygge bro mellom grunnforskning og klinikk. Dette er viktig anvendt forskning til det beste for samfunnet. Ved å samle forskere på tvers av fagområder vil Jebsen-senteret etablere et nettverk av forskere for å fremme vitenskapelig diskurs og for å gi samfunnet kvalitetssikrede vitenskapelige fakta om Alzheimers sykdom og om hjernen. Dette senteret vil bidra med forskning til en sykdom som preger samfunnet vårt, og som er etterspurt i å forstå. Jeg vil igjen gratulere så mye med dette senteret, og jeg gleder meg veldig til å følge forskningen og de resultatene som kommer ut av senteret. Jeg vet at dere klarer å frambringe fremragende forskning.
Jebsen-senter for Alzheimers sykdom har en fantastisk visjon. Taking the Nobel Prize to the clinic. Og for meg er det enkelt oversatt at den fremragende forskningen som er gjort på dette området skal nå komme enda mer pasienter, pårørende og samfunnet til gode. For hvem av oss er vel ikke berørt av denne sykdommen? Vi har venner, familie, alle kjenner vi noen som har den. Og dette senteret er et uttrykk for hvordan vi jobber i det integrerte universitetssykehuset. Fremragende forskere både fra NTNU og fra St. Olav ved nevroklinikken jobber sammen for et felles prosjekt. Jeg ønsker dere lykke til.
Før vi går videre i programmet, så har jeg tenkt å ta på meg den litt høye legehatten min. For det er nemlig sånn at du som sitter og ser på nå, har et sted mellom 10-20% sannsynlighet, eller risiko, burde jeg si, for å utvikle Alzheimers sykdom. Men det er faktisk ikke sånn at du da bare skal lene deg tilbake og la det som skjer, skje. For det viser seg nemlig at forskerne har funnet ut at det er opp til 12 risikofaktorer for å få demens som vi kan gjøre noe med. Og innad i de 12 tinga så er det selvfølgelig sånn at det ligger dette med livsstil. Og det betyr da at selv om jeg kan være den første til å innrømme at det er vanskelig å gjøre livsstilsendringer, så er det jo litt deilig å vite det at det finnes faktisk ting vi kan gjøre for å ta vare på hjernehelsa vår. Og blant de tinga så vet vi for eksempel at det gjelder å slutte å røyke. Vi må holde litt kontroll på blodtrykket vårt, og en av de gøyale tingene som man faktisk kan gjøre noe med, det er å være mer sosial. Så vil jeg jo si da at en av de store visjonene bak skapelsen av det nye senteret vårt, det har vært å skape samarbeid på tvers av fagdisipliner. Og det er jo sånn at vi alle sammen har et mål om å nå nettopp det å knekke den store Alzheimer-koden. Men hvordan er det egentlig forskerne på det nye Jebsen-senteret har tenkt til å jobbe mot et så hårete mål? Det vi har gjort, det er å sette sammen en kort oversikt om hva det er forskerne skal drive med her på senteret, og det skal dere få lov til å se på nå.
I 2014 så fikk vi Nobelpris på grunn av at vi hadde gjort store funn når det gjelder stedsansen. Og nå feirer vi at vi kan ta disse funnene her med oss ut i klinikken for å prøve å forstå hva er det som skjer i Alzheimers sykdom. Så jeg representerer den første arbeidspakken, der vi er interessert i å finne ut av celler som vi har da oppdaget hos dyr, som er viktig for stedsansen. Og før så hadde vi gittercellene, og nå har vi også funnet objektvektorceller. Det høres veldig komplisert ut, men det er det ikke. Det er bare celler som reagerer på ting som er ute i rommet, med å måle både avstand og retning til deg, og signalisere det.
I en del av hjernen, som kalles den entorhinale cortex, har vi en type hjerneceller som hjelper oss med å finne fram. Disse cellene kalles gridceller, eller gitterceller, og de danner et koordinatsystem, eller et slags stabilt mentalt kart over omgivelsene våre. Dette kartet hjelper oss for eksempel med å kunne ta snarveier, og det hjelper oss også med hukommelsen. For eksempel så kan du se for deg at du sitter på stua, og så går du ut på kjøkkenet for å hente en ting. Men i det du kommer ut på kjøkkenet, så har du glemt hva det var du skulle hente. Da kan det hjelpe å gå tilbake til stua, altså til det stedet du var da du tenkte denne tingen, fordi minnet om tingen er knyttet til stedet, altså til stua. Gridceller ble først oppdaget i rottehjerne, og senere også i musehjerne. Og selv om det er store forskjeller mellom oss og rotter og mus, så har man også funnet gridceller i menneskehjernen. Man regner med at de finnes hos alle pattedyr. Hjerneceller kommuniserer ved hjelp av elektriske signaler, som vi kan lytte til og se på labben. Og ved å lytte til disse signalene i rotter og mus, så kan vi finne ut hvordan cellene fungerer i normale, friske hjerner.
I daglig gjøremål så er vi nødt til å interagere med ulike objekt. Det kan være personer, det kan være bord, stoler, bygninger, biler osv. Når vi skal interagere med disse objektene, og når vi skal ta oss fram i verden som inneholder disse objektene her, så er vi nødt til å ha en forståing av den romlige relasjonen mellom oss selv og de objektene. For eksempel når vi skal navigere, så kan vi bruke retning og avstand til et kjent landemerke, for eksempel en stor bygning, til å finne fram til målet vårt. Når vi skal spille fotball, så er vi nødt til å ha en anelse om retning og avstand til medspillere og motspillere, og hvordan det relaterer til sidelinjene og målene osv. Og nå tror vi at vi vet hvordan hjernen er i stand til å kode slike romlige relasjoner til objekt. I et område i hjernen som heter mediale entorhinal cortex, så finner vi en viss type nerveceller som heter objektvektorceller, som koder retning og avstand til spesifikke objekt i omgivelsene. En enkelt objektvektorcelle spesialiserer seg på å kode en viss retning og en viss avstand til et objekt. For eksempel kan den bli aktiv når dyret er halvmeter rundt et objekt og øst for det. Andre objektvektorceller spesialiserer seg på andre retninger og andre avstander. Som en populasjon så dekker objektvektorcellene alle retninger og avstander. Sånn at ved å lytte til hva slags celler som er aktive, så kan du gjerne alltid vite hva er retningen og hva er avstanden til et visst objekt. Og vi tenker oss at objektvektorcellene og gittercellene de jobber sammen. Sånn at for eksempel når du spiller fotball, så kan gittercellene fortelle deg hva du er hen på banen, mens objektvektorcellene forteller deg retningen og avstanden til medspillere og motspillere på den banen.
Og vi har tenkt å finne mye mer ut av hvordan disse her fungerer i dyra våre. Og grunnen til vi er interessert i det, er også for å prøve å forstå, finnes det muligheter for det vi lærer hos dyra, å ta med oss ut i den normale hjernen hos mennesker, og spesielt i den hjernen der disse cellene her blir borte. Hvilke funksjoner blir da borte ved siden av stedsansen? En av de syke hjernene som vi vet får problem med stedsansen, det er når en får Alzheimers sykdom. Da vet en at disse cellene her dør. En kan se det på MR at entorhinal cortex, basen for disse her cellene som vi nettopp har beskrevet, den skrumper fordi cellene dør. Og det betyr da at pasienter som begynner å få problem med å finne fram, med å orientere seg i miljøet til objekt og til andre ting, det er tegn på Alzheimers sykdom.
Så formålet med arbeidspakke 2 er å finne ut hvordan hjernen holder styr på tid. Når vi opplever hendelser, så skjer det i en bestemt rekkefølge, og det må hjernen prøve å sortere ut. Vi tror at en god del av det skjer i et område som vi kaller lateral entorhinal cortex, ofte så sier vi bare LSC. Det vi ønsker å finne ut i denne arbeidspakken, det er hvordan skjer det i LSC, hvordan skjer det i samarbeid mellom mange nerveceller, og hvordan blir det ført videre til et annet hjerneområde, hippocampus, som er veldig viktig for hukommelse. Da tror jeg at vi vil forstå hvordan tid, eller episodisk tid, oppstår, altså rekkefølge av hendelser, hvor det blir satt sammen, og hvordan det blir lagt ned i hukommelsen for dagligdagse hendelser.
Når vi snakker om tidssansen i den konteksten her, så snakker vi om det vi kaller episodisk tid. Og episodisk tid, det er en helt essensiell del av det vi kaller episodisk hukommelse, som er minner for egenerfarte situasjoner. Og slike minner de kan vi typisk dele inn i hva som skjedde, hvor det som skjedde, og når det skjedde. Og det er nettopp denne når-komponenten av de minnene vi kaller episodisk tid. Og for å kunne gi et godt eksempel på at den formen for tid ikke er det vi kaller absolutt klokketid, men heller en form for subjektiv tid, så kan jeg ta en opplevelse og stille et spørsmål om for eksempel, når kjøpte du matvarene dine sist? Og hjernen din gjør ikke noe sånn som at den svarer da, det må ha vært mandag 16. november klokken 18.03. Det den heller gjør, er sannsynligvis at den går igjennom hvorvidt det kan ha vært i går, og det kan det ikke, fordi da var du på middagsselskap hos en venninne. Og det kan heller ikke ha vært dagen før det igjen, fordi da jobbet du så sent at du rakk ikke å spise middag en gang. Så det må faktisk ha vært mandagen, for da sluttet du nemlig tidlig på jobb og kunne lage den middagen. Så det du ser at hjernen din har gjort, er at den tar hendelsene du har erfart, og så organiserer den det i en meningsfull rekkefølge. Og den måten å kode tid på, det vet vi som sagt er en helt essensiell del av hukommelsen vår for erfaringer vi har gjort oss. Og den typen hukommelse, den bor i senteret som kalles hippocampus. Det som er inngangsporten til hippocampus, det kaller vi entorhinal cortex. Og fra før av så vet vi at den mediale delen av entorhinal cortex, den koder for stedsansen. Og da kan man godt tenke seg at mediale entorhinal cortex sender informasjon til hukommelsessenteret vårt om hvor-komponenten av de episodiske minnene våre. Mens den søsterregionen som vi kaller laterale entorhinal cortex, den har det vist seg at faktisk koder for nettopp en sånn form for subjektiv episodisk tid. Det vi da kan forestille oss at vi har hukommelsessenteret vårt hippocampus som integrerer nettopp den informasjonen om hvor noe skjedde, når noe skjedde, og hva det som faktisk skjedde.
Den tidssansen, eller uttrykket for forløp av tid, det oppdaget vi for et par år siden at veldig avhengig av et hjerneområde som heter lateral entorhinal cortex, LEC som vi kaller det. Nå det sånn at det det samme området som ofte det aller første til å bli rammet i Alzheimers sykdom. Ofte så begynner celledøden i hjernebarken akkurat der, og så sprer det seg videre til de spatiale cellene, eller gridcellene, og til hippocampus for hukommelsessystemet. Det betyr at det veldig viktig å finne ut hva som går galt i det LEC-området. Det noe som vi skal gjøre da, både i studier på rotte og mus i arbeidspakke 2, men også i studier på mennesker, der vi i arbeidspakke 4 spesielt vil se på samme området i menneskehjernen, og spesielt i hjernen til mennesker som har fått, eller i en tidlig fase av Alzheimer, eller kanskje enda til bare en risikosone for å få Alzheimer. Og da spør vi da, det sånn at tidssansen, eller den episodiske tidssansen, evnen til å se ting i rekkefølge, forstå rekkefølge og huske ting i riktig rekkefølge, om det også rammet når disse cellene dør såpass tidlig. Og når jeg sier såpass tidlig, så det vel grunn til å tro at den prosessen kan starte ikke bare mange år, kanskje enda til 10 år, før vi får kliniske symptom på Alzheimers sykdom.
Vi vet nå at den hjernedelen som rammes først i Alzheimers sykdom, lik imellom mennesker og andre pattedyr, inkludert rotter. Det gjør at vi kan bruke rotter som utvikler en form for Alzheimers sykdom i forskningsarbeidet vårt. Dette veldig verdifullt, fordi rotter eldes mye raskere enn mennesker. De gamle når de om lag 1,5 år. Det gjør at vi kan studere prosesser i Alzheimers sykdom langt mer effektivt ved bruk av rotter enn menneskehjernen. I tillegg kan vi gjøre studier på en rottehjerne som langt mer dyptgående enn det som mulig i en menneskehjerne. Vi kan for eksempel ta ut hjerneceller, sårbare hjerneceller, fra en rotte når rotten dør, men allikevel holde cellene i live. Slik kan vi studere disse sårbare hjernecellene i en liten laboratorieskål. Det muliggjør en veldig direkte type studie av disse cellene. Vi kan også studere cellene i rottehjernen mens de i live, og bruke egenutviklede mikrobiologiske verktøy for å holde disse cellene friske. Dette muliggjør et nivå på studien av Alzheimers sykdom som ikke tidligere har vært mulig. I arbeidspakke 3 skal vi forsøke å finne ut mer om hva som forandrer seg inne i disse sårbare hjernecellene. Det vil si hvilke små, men avgjørende endringer som fører til Alzheimers sykdom. Ved å forandre på mengden av ulike proteiner i de hjernecellene som rammes tidlig ved Alzheimers sykdom, håper vi å kunne finne ut hvilke endringer det her igjen medfører. Gjennom denne tilnærmingen så håper vi å kunne avdekke mer av hvilke forandringer som oppstår helt tidlig i sykdomsforløpet ved Alzheimers sykdom. Gjennom å bruke disse metodene så håper vi å avdekke hva som skal til for å kunne utvikle en kur mot Alzheimers sykdom.
Når vi jobber med de spennende celletypene som vi finner hos rotter, slik som gridceller og objektvektorceller, så lurer vi jo veldig på om dette relevant for mennesker. Og om det vi finner ut kan bidra til å forstå hjernesykdommer. Særlig i møte med pasienter. Det jo flere av oss som jobber på Kavli Institute, som også utdannet som leger. På den ene siden så det jo mye som felles mellom nerveceller hos rotter og mennesker, slik at det rimelig å tro at mye av det vi finner vil være relevant for mennesker. Men på den annen side så det jo mye som forskjellig fra grunnforskningen på Kavli Institute og klinisk behandling av hjernen på sykehuset. Ikke bare rotter og mennesker tross alt ganske forskjellige, men også måten vi kan studere dem på ulik. Og det ikke sikkert at det vi synes spennende i grunnforskningen relevant i klinikken. Men det kan jo være det, og det akkurat det vi må prøve å finne ut. Og det er en vanskelig oppgave som krever at forskere med helt ulik bakgrunn klarer å jobbe godt sammen. Gjennom Jebsen-senteret så prøver vi å gjøre nettopp det, å bygge denne broen mellom grunnforskning og klinikk, steg for steg. I prosjekt 4 fokuserer vi på 2 viktige steg i denne translasjonen. Først så ønsker vi å undersøke om funnene fra Moser-gruppa, om hvordan tid og sted prosesseres i hjernen, også gjelder for mennesker. Vi kan ikke studere dette på samme måte i mennesker som vi kan i rotter, så her bygger vi på en kombinasjon av psykologifeltet og funksjonell avbildning av hjernen, altså fMRI. Med denne teknikken undersøker vi aktiviteten i spesifikke hjerneområder og ser på hvordan denne aktiviteten endrer seg i forhold til det vi gjør, slik som å navigere i omgivelsene. Men siden man ikke kan navigere rundt i ekte omgivelser når man ligger i MR-skanneren, så må vi gjøre det på en litt annen måte. Det vi gjør er å bruke dataspillignende teknologi, slik at deltakerne kan i stedet navigere i en virtuell virkelighet. Deretter undersøker vi om hjerneaktiviteten endrer seg i forhold til hvordan man navigerer, og, viktig poeng, om dette skjer på en lignende måte som vi ser hos rottene. Og på den måten så kan vi undersøke både forskjeller og likheter i hvordan hjernen prosesserer tid og sted hos rotter og mennesker. I neste steg ønsker vi å finne ut om disse prosessene påvirkes av Alzheimers sykdom. Og for å finne ut det, så jobber vi tett sammen med nevrologisk avdeling på Olavs hospital i prosjekt 5 i Jebsen, og rekrutterer da både pasienter med tidlig Alzheimers sykdom og friske frivillige i samme aldersgruppe. Det som er så spennende med denne tilnærmingen, er at vi håper å kunne knytte funnene våre både mot basalforskningen om tid og sted, og mot klinikken. Og i tillegg mot forskning på biokjemiske markører, som også foregår i Jebsen-senteret. Og på den måten håper vi å kunne bygge en bro, slik at forskningen på stedsans og tidssans kan bidra til å forstå hjernesykdommer.
Her på nevrologisk avdeling ved Olavs hospital har vi ansvar for den delen av Jebsen-senteret som handler om pasienter. Vi har organisert arbeidet i forskningsgruppa Trønderbrains, som ble startet allerede i 2003 som et samarbeid mellom Olavs hospital og NTNU. Her arbeider vi med å forstå hvordan nevrodegenerative sykdommer oppstår. Nevrodegenerative sykdommer er sykdommer hvor nerveceller gradvis dør, og Alzheimers sykdom er den viktigste av de sykdommene. Hvis du eller en av dine nærmeste skulle få problemer med orientering eller hukommelse, eller andre problemer med det vi kaller kognitive funksjoner, og blir henvist til Olavs hospital, så er det til oss du kommer. Hos oss i Trønderbrain-gruppa ved nevrologisk avdeling, så har vi laget et spesielt system for hvordan vi utreder slike pasienter. Vi bruker ulike spørreskjemaer for å kartlegge det som kalles den kognitive funksjonen. Og i tillegg gjør vi alltid spinalvæskeundersøkelser og en MR av hjernen. De friske, frivillige kontrollpersonene som vi også ønsker å ha med, de undersøker vi på akkurat samme måte. Vi jobber med å forstå hvordan Alzheimers sykdom oppstår. Hvorfor dør nerveceller i visse hjerneområder? Det er det viktigste spørsmålet vi stiller oss i Jebsen-senteret. I tillegg vil vi i Trønderbrain-gruppen også være veldig opptatt av å finne gode markører som kan sette oss i stand til å stille Alzheimer-diagnosen så korrekt og så tidlig i forløpet som mulig. Vi tror at vi ikke får tilbake nerveceller som allerede er døde. Derfor blir det veldig viktig å klare å stanse sykdomsutviklingen så tidlig som mulig. Det er særlig viktig den dagen vi forhåpentligvis får en effektiv medisin som vi kan tilby pasientene våre. Gjennom Trønderbrain-prosjektet har vi samlet blodprøver fra mer enn 1000 pasienter og 1000 friske frivillige til genetisk undersøkelse. Fra 2008 har vi også samlet spinalvæske på pasientene, og vi har gjort MR-undersøkelse av hjernen av alle sammen. Dette har til sammen gitt oss et fantastisk spennende forskningsmateriale som er attraktivt å dele med forskningsgrupper. Og vi samarbeider derfor med mange andre forskningsgrupper, både i inn- og utland. I Jebsen-senteret vil bidraget vårt særlig være å rekruttere pasienter med Alzheimers sykdom i tidlig fase og friske frivillige. De skal gjennomføre avanserte MR-undersøkelser. Målet er å vise at funnene som ble gjort hos rotter og som førte til Nobelprisen, også er gyldige i menneskehjernen. Vi er allerede i gang med funksjonelle MR-undersøkelser. Det vil si MR-undersøkelser som kartlegger funksjon og aktivitet i visse hjerneområder. Det gjør vi gjennom et dataspill-lignende oppsett, hvor pasientene og de friske frivillige orienterer seg i et virtuelt rom mens de ligger i MR-maskinen. Nylig satte vi i drift den nye og svært avanserte 7 Tesla MR-maskinen, som tar spesielt fine bilder av de hjerneområdene vi er interessert i å studere. Innen 1 til 2 år får vi også PET-metoder som kan studere de samme hjerneområdene med vekt på funksjon ned på molekylært nivå. Vår egen forskning, som går på å kunne stille diagnosen Alzheimers sykdom sikkert og tidlig, kommer til å fortsette som før. Vi er opptatt av å finne gode metoder hvor vi kan analysere prøver fra spytt, eller blod, eller spinalvæske. Er du frisk mellom 55 og 80 år og interessert i å bidra som frivillig i vårt prosjekt? Da kan du gjerne ta kontakt med oss.
Vi er ikke så vant til gode nyheter, vi som jobber med demens. 100 år med forskning uten gjennombrudd og uten at noen har funnet en kur, kan gjøre noe med optimismen og troa. Men nå begynner det å skje noe. Det går noen rotter rundt på gulvet i et laboratorium i Trondheim med målere på hodet. Og ved hjelp av de har vi kommet mye nærmere å finne startpunktet hvor demenssykdommen begynner. Og klarer vi å finne det startpunktet, kan vi også finne stoppunktet. Vi får nyheter fra Sverige om en blodprøve som kan gi diagnose på demens mye før den synes for alle andre. Hvis vi får til dette, betyr det at vi er klare til å sette i gang behandling når en kur finnes, mye før skaden er for stor på hjernen. Det vil også bety mye. Og i USA så er det nå en ny vaksine på gang, som vi har forhåpninger til. Den kan ikke stoppe sykdommen, men den kan gi en kraftig forsinkelse. Og for pasienter og pårørende er de ekstra årene man kan få da, en veldig verdifull gave. Nobelprisen til Moser-teamet i 2014 ble en viktig begynnelse. Det var med på å gi forskningen økt interesse, økt status, og med det økte investeringer. For også her handler det om penger for å kunne holde trykk oppe, for å kunne forske, for å kunne finne fram til løsninger og gjennombrudd. Derfor er vi så glade for at det er mange nå som går sammen for å være med på denne jakten i letingen på en kur. Og derfor er vi så glade for at KG Jebsen-senter for Alzheimers sykdom nå etableres. Finansieringen fra stiftelsen Christian Gerhard Jebsen har gjort dette senteret mulig, sammen med støtten fra Helse Midt og NTNU. Det betyr veldig mye for pasienter og pårørende, og det satses så mye på forskning innen demens. Vi i Nasjonalforeningen Folkehelsen har vært med i Moser-nettverket fra start, gjennom vårt demensforskningsprogram. Vi kan bidra med brukermedvirkning, slik at stemmen til de dette faktisk handler om, de som har skoa på, de som er rammet, er med på å gi innspill i hele forskningsprosessen. På vegne av Nasjonalforeningen Folkehelsen er jeg utrolig stolt av og glad for at vi også er en del av samarbeidet, der forskere, akademia, klinikere og vi, pasientene og pårørende, er involvert i et bredt samarbeid for å løse gåtene. Åpningen av dette senteret er med på å gi optimisme, det er med på å gi håp. Gratulerer med åpning og oppstart. Vi gleder oss til fortsettelsen.
Jeg vil gratulere oss alle med dette nye forskningssenteret, KG Jebsen-senteret for Alzheimers sykdom. Vi trenger mer kunnskap om Alzheimers sykdom, og her skal man kombinere nobelprisvinnende forskning på hukommelse og stedsans med det å få til en god klinisk forskning rundt Alzheimers sykdom, prøve å få til bedre diagnostikk og behandling. Det viser jo seg at noe av de første symptomene man har ved Alzheimer går jo på det med forståelse av tid, stedsans, så da det er en kobling her som vi tror kan bli veldig viktig til beste for pasientene. Og de som rammes jo hardt av denne sykdommen, ikke bare pasientene, men familien og samfunnet for øvrig og helsetjenesten. Så vi trenger virkelig en kjempeinnsats mot Alzheimers sykdom, og jeg har stor tro på at dette senteret skal bidra til det.
Kjære Edvard, May-Britt og øvrige kolleger ved KG Jebsen-senter for Alzheimers sykdom. Jeg vil først og fremst få gratulere dere med en ærefull tildeling og nok en fortjenestefull anerkjennelse av det fremragende forskningsarbeidet som utføres ved Kavli-senteret. Det vil alltid være et viktig mål for medisinsk basalforskning å kunne ta dette videre til klinisk anvendelse. Og derfor ekstra gledelig at basalforskere med Nobelpris på hjernens indre GPS hos rotter blir tildelt status som et fremragende senter for translasjonsmedisin. Jeg merket meg også at Edvard selv i en uttalelse til NRK omtaler det som en stor lettelse at dere nå er i stand til å ta forskningen fra labbenken til sengeposten. Som et Jebsen-senter for genetisk epidemiologi har vi gått veien fra befolkningsstudier som hund til laboratoriebenken og til klinisk translasjon. Gjennom omfattende genetiske analyser av 90 000 trøndere har vi et fantastisk presisjonsmedisinsk verktøy for å studere genetisk risiko for en rekke ulike sykdommer. Dette er et svært spennende utgangspunkt for samarbeid mellom våre to Jebsen-sentre her ved NTNU. Nettopp å se etter endringer i entorhinal cortex hos eldre personer i hund ved hjelp av funksjonell MRI, orienteringsevne, kognitive tester og genetisk risiko. Vi vil nok en gang få gratulere med dagen, og vi ser fram til et meget interessant forskningssamarbeid mellom to gode naboer. Takk.
Good morning, everybody. My name is Hans-Peter Jebsen, and I represent Stiftelsen Jebsen. I wish to take a few moments to give you a very brief introduction to Stiftelsen and what we do. Stiftelsen Jebsen was founded in 2009 to honor my father, Christian Gerhard Jebsen, his name, contribution to Norwegian shipping and business, and to give something back to the society. Since the start, Stiftelsen has been engaged in maritime projects, cultural projects, and not the least medical research. The latter is by far the biggest area where we, since 2011, have contributed a total of about 470 million kroners to help establish 22 KG Jebsen centers for medical research, medicine, and affiliated university hospitals. All the KG Jebsen centers must be based on translational research and hopefully leading to improved patient treatment in the not too distant future. Today there are 10 active KG Jebsen centers out of the 22 that have been established since 2011. Each center has been or will be funded for a period of 4 to 6 years. As to follow-up, we have our Medical Advisory Committee. They evaluate the midterm report for each center, in addition to the annual report on status of research and finances. To rate and evaluate our KG Jebsen Center program, we have twice, in 2014 and 2018, had an external international expert panel give us feedback on the program, what has been good and where improvements can be made. On both occasions, the conclusion has been that the program has given the intended results. It has made a difference. So back to why we are digitally here today, the official opening of KG Jebsen Center for Alzheimer's disease. You will all talk about the medical aspects, but I can only say that Stiftelsen Christian Gerhard Jebsen is proud to be part of a possible solution to Alzheimer's disease, and as you say, take Nobel Prize to the clinic. Please make a difference, and good luck, and thank you.
Da har vi kommet til det som noe av dagens store høydepunkter. Vi skal nemlig få høre fra internasjonale eksperter om Alzheimers sykdom rett fra forskningsfronten. Men jeg vil også understreke her at det viktig at dere nå engasjerer dere, for dere har faktisk muligheten til å ha direkte kontakt med Alzheimer-forskerne våre. Og det dere gjør da igjen, det at dere bruker Slido-appen, og ikke bare nok med å kunne sende spørsmål til forskerne deres, så kan dere, som ikke nødvendigvis brenner inne med spørsmål selv, gå inn og stemme fram spørsmål dere synes vi skal få svar på, og på den måten forme programmet for dagen. Og før vi går videre, så vil jeg bare si det at vi vet at hukommelsen vår avhengig av at hjernecellene, de snakker sammen. Og kommunikasjonen mellom hjernecellene våre, den foregår i det vi kaller synapser, som kontaktpunkter som cellene danner mellom hverandre. Og den første foreleseren ut i dag, som skal holde et foredrag for dere, det Tara Spires-Jones. Hun professor ved Universitetet i Edinburgh, og i tillegg til å gi oss en kort oversikt over det vi vet om Alzheimer fra før, så skal hun fortelle hvordan hennes forskningsgruppe forsøker å kartlegge hva det som forårsaker at nettopp disse kontaktpunktene mellom cellene ser ut til å forringe hos pasienter med Alzheimers sykdom. I tillegg så skal Tara fortelle litt om hva hun tenker veien videre for å finne gode behandlingsmetoder. Så da sier vi bare, vær så god til Tara.
Hello everyone. Thank you very much for the invitation to give this lecture. It's my honor to be here today. What I'll talk about is Through the Looking Glass, imaging brain changes to future life-changing treatments for dementias. And I talk about Through the Looking Glass because much of what we do in the lab is looking down microscopes, looking through glass to see what changes in the brain. And what I'll start with today is a little bit of talk about the past, what we already know about the brain and Alzheimer's disease from prior research. I'll talk about the present, a little bit of the current research in my group at the University of Edinburgh, and then I'll end with a few words about the future and our path to effective treatments based on research. But first, a little bit of introduction and the past. What do we know about the brain? Well, the first thing that I can share with you about the brain is it's absolutely amazing. Your brain contains about 100 billion neurons, or the brain cells that do the talking to each other in the network. They're connected to each other through 100 trillion synaptic connections. That's tons, that's lots and lots of synaptic connections. And if you look in detail, here is an image of the brain, and we can see it from the side here. And you can also see, if we cut through the brain, a coronal section, you can see the gray matter on the outside where these 100 billion neurons or brain cells are located, and the white matter running through the inside of the brain, and this is the wires that connect the different parts of the brain together. And if we zoom in, we can see beautiful neurons. These are drawings by Santiago Ramon y Cajal, who looked down the microscope for the first time at brain cells stained with silver stain. And he described the neuron cell bodies, the beautiful dendrites that receive connections, and the axons that send connections. And that point that I told you about, the connections, are called synapses. And here I'm showing you an image with a transmission electron microscope of a single synapse. So this is the very end of an axon with lots of little vesicles containing neurotransmitter. And when an electrical signal comes down the wires through the white matter, it reaches this synapse, it releases the neurotransmitter, which then binds receptors in the postsynaptic density and starts an electric signal in the next neuron down the train. And those 100 trillion synapses means that within your brain, there are more synapses than there are stars in our galaxy. And this is one of the reasons we don't have effective treatments yet for dementias, and that's because the brain is phenomenally complex, and we don't fully understand how the brain works when it's healthy, so it's taking us a long time to understand how it goes wrong in disease and how to treat it. Why do we study Alzheimer's disease in my lab? I think you probably will all realize that this is a really devastating disease. And what I'm showing you here are 3 examples. This is my grandma, Natane Robertson, who died in 2007, and my Mima, my other grandmother who is still alive today. And they have both lived long and healthy lives without any cognitive decline. But this lovely woman here, Joan Jones, my grandmother-in-law, she died with Alzheimer's disease. And it's a very devastating condition. And not only is it devastating for people living with it and their families, but it's a massive problem in the world. These data I'm showing you here are from 2015, which indicated about 9 million people in the Americas had dementia at the time, 10.5 million in Europe. And the estimates worldwide now are that 50 million people are living with dementia. This costs about 800 billion euros to care for them every year for everyone who has dementia. And right now, to make it worse, we don't have any effective treatments to stop the progression of the disease, but research will lead to disease-modifying treatments, or hopefully also preventions for the diseases that cause dementia. So what causes dementias like Alzheimer's disease? Alzheimer's is one of the causes of dementia, and it's the most common. It causes about 60% of the people with dementia. And we know that dementia's risk is influenced by aging, lifestyle factors, and genes. And this is very important because we can use genes to understand the biological changes in the brain, and we can also think about lifestyle factors that we can all change to reduce our own risk of dementias. It's estimated that over 1/3 of dementias could be prevented by changing modifiable or lifestyle risk factors. Things that are associated with increasing dementia risk, which are bad for your brain, are smoking, high blood pressure, obesity, diabetes, physical inactivity, social isolation, depression, and hearing loss. And a new one that's been discovered lately is brain injury, traumatic brain injury. Things that are protective against dementia or associated with a lower dementia risk are exercise, healthy diet, a higher level of education, and keeping physically and socially active. But we don't fully understand how these things influence dementia risk, or even if some of these things that are associated with dementia risk, for example, depression, whether those cause dementia or whether those are a symptom of the early stages of dementia before we even know the disease is present. So one of the things we're doing in the lab is trying to bridge this key knowledge gap, understanding the biological changes in the brain that link these risk factors and increased risk of disease. Some of the changes that we know about in the brain are these changes in your plaques, tangles, synapses, and neurons, and I'll give you some pictures of these later. But what I want to show you here is the pathological changes in the brain, including these amyloid plaques, which clump up abnormally in the brain. They're occurring and ramping up in the brain for many years in what we call a prodromal phase. That is when you don't have any detectable disease symptoms. This is sort of the amount measured in the brain. And you see, by the time you have a diagnosis of mild cognitive impairment, you already have a brain pretty much full of an amyloid plaque protein. There's also accumulation of tau and tangles, and I'll show you a picture in a minute. But what you'll see is the tau tangles start to accumulate in this prodromal phase, but they really don't spread through the brain and accumulate in large amounts until you have a symptom onset, when you have this mild cognitive impairment. And we see that the loss of the neurons, the brain cells, and the loss of the synapses sort of occur over many, many years, which is another reason we don't yet have effective treatments for these diseases, is that there's a very long period where people have the brain changes but don't yet have symptoms. So we need to be able to treat these people very early and have markers, like Professor Zetterberg will talk about, of brain changes early on. But of all the brain changes that we see in Alzheimer's disease, the loss of these tiny synaptic connections that I told you about is the strongest pathological correlate of the symptoms of dementia. So these are what we work on in my lab that I'll tell you a little bit about over the next 10 or 15 minutes. And here's an example of these brain changes. The plaques, tangles, and atrophy, which are the 3 defining features of Alzheimer's disease, were first described in the early 1900s, in 1906 and 1907, by this person, Dr. Alois Alzheimer. And he described them about his patient, August Dieter, who came to him and said, "I've lost myself." She had the devastating memory loss and changes in her brain that we associate with Alzheimer's disease today. And what Alzheimer found when he looked at this person's brain when she died was these 3 things. Atrophy, and you can see that here, that means loss of brain material. This is a brain section, coronal section through someone who died of non-neurological causes. And this is a similar section from someone who died with Alzheimer's disease. And you can see in particular this little part of the brain here called the hippocampus is very, very shrunken compared to in the control. And the hippocampus is very important for learning and making new memories. Also, the lovely curves on the outside of the brain, the neocortex, is very shrunken. And that's very important for thinking and memory as well. And when Alzheimer looked down a microscope, very similar to what Cahal had done to look at neurons, he saw amyloid plaques, he called them senile plaques, and neurofibrillary tangles. And we know now that these are made of that amyloid beta peptide, the plaques, and the tangles are made of a tau protein that's gone to the wrong place in the cell and they clump up abnormally. But it's not just the neurons and the plaques and the tangles that go wrong in the brain. There's also inflammation. So there are other cell types called astrocytes and microglia that clump around plaques. They come and try to clear these abnormal toxic clumps of protein, but they themselves, as they try and benefit the brain, then become a little bit toxic. They get too inflamed and secrete things that damage the surrounding neurons and synapses. So that's the background. Now I'll talk for a few minutes about what we're doing in our lab at the University of Edinburgh in the UK Dementia Research Institute to understand some of these brain changes. So in the background that I told you about, in the field, we understand that there are risk factors, age, genes, and lifestyle, like I talked about. And then early on, these lead to amyloid, or this plaque accumulation. And a little bit later on, and we think downstream, this leads to the tangle or tau accumulation. And this then leads to the neuron loss, the synapse loss, and the terrible symptoms of the disease. We're trying to understand what causes the synapse loss that correlates very closely with cognitive decline, and can we prevent or even reverse the synapse loss in the brain? And what our research has suggested is a little bit more of a complicated cascade than this classic amyloid cascade that people talk about in the field. I'll tell you a little bit about two of the genes that we're studying, ApoE and clusterin, and I'll talk a little bit also about age and lifestyle, and how we think these risk factors act through glia, which are these non-neuronal cell types in the brain that become inflamed, to mediate the A-beta and tau-associated synapse degeneration and cognitive impairment. And what we are finding in the lab is that A-beta and tau contribute to synapse dysfunction and loss, and that at least in animal models, these changes can actually be reversed. How do we answer these kind of questions? We use multiple different model systems and techniques in the lab. We look at human brain that's been very generously donated post-mortem after people died with dementias, and control brains. We use animal models, including flies and mice, and we use cells in dishes, cells derived from human brain and skin cells, and from mouse models as well. What we've found recently is that the plaque protein, amyloid beta, that clumps up abnormally in the brain, the amyloid plaques are surrounded by a halo of smaller clumps of amyloid called oligomers. And when we look really closely with techniques we pioneered in the lab called array tomography, we can see inside individual synaptic terminals, both the presynaptic side and the postsynaptic side, so the side that's sending the message and the side that's receiving the message, both of these have small clumps of amyloid in the synapses, and they have more of this as you get closer and closer to the plaque. So this graph is showing you the percentage of synapses that contain these toxic oligomeric clumps, and this is showing you the distance from the edge of a plaque, and you can see as we get closer and closer to the plaque, we have more and more of the synapses that contain this toxic A-beta. And this first set of data was collected from mouse models expressing a gene that causes Alzheimer's disease, and they develop plaques. But we were also able to use this same technique and adapt it for use in human brain tissue. And what we found is similarly, we have clumps of A-beta inside synapses. We have loss of synapses as we get closer to plaque. So this is the density of these important synapses, and this is the distance from the plaque on the x-axis. And you can see as we get closer and closer to the plaque, we have fewer and fewer of these synaptic connections. We see the oligomeric A-beta clumping inside. These are microscope images showing you blue is the plaque protein, red is actually a risk gene called ApoE, and green is a synaptic protein. And what we see is in people who carry the ApoE4 version of this gene, which increases your risk of Alzheimer's disease, have 5 times more synapses with the toxic A-beta peptide compared to people who have the ApoE3 version of the gene. So this is linking one of the biggest risk factors, one of the risk genes for Alzheimer's disease, with clumps of toxic oligomeric A-beta inside synapses where we think it's damaging and killing the synapses. Recently, we've linked another risk gene called clusterin to increased clumping of A-beta in synapses in AD. And I'm showing you here, this time the plaque protein is in cyan, and the presynaptic terminals are in yellow, and the clusterin, which is another risk protein, is in magenta. And we see clumps of this clusterin in the synapses in Alzheimer's disease brain. We see it more in Alzheimer's than control, and we see it more in the people with the risk variant of ApoE4. So now we've linked 2 risk genes for Alzheimer's disease with clumping of A-beta inside synapses. And this is really interesting because these risk genes are found in the brain, not only in the neuron cells that do the thinking, but they're found in astrocytes and microglia, those 2 types of support cells that I was talking about that also accumulate around plaques. And in fact, in this image, you can see clusterin staining a cell that's probably not a neuron, that's probably an astrocyte here, just right next to the plaque. And what this made us think is we know from genetic studies that other people have done that ApoE and clusterin are risk genes, and we also know that they are upregulated, or they're expressed more inside glia when you have the disease, and especially around plaques. So we thought that maybe the glia are involved in this synapse pruning or this synapse degeneration process. And we've been looking at that recently in the lab. This is a PhD student, Makis Sioras, who has found that in Alzheimer's disease brain, he sees more synapses inside microglia than in controls. So what we're showing you here in green are the little synaptic puncta in a human brain, and in pink or magenta are lysosomes, or a part of a microglial cell, which is one of the glia, and in blue are plaques and the nuclei of cells. And in the Alzheimer's brain, you can see more of this pink, more of these microglia clumping around plaques, and inside of them, you can see the green protein is accumulating. So this is telling us that microglia are ingesting synapses in human AD brain, and we looked in 2 parts of the brain, one in the temporal lobe, which is heavily affected by pathology, and one in the occipital lobe at the back of the brain. And in both areas, we see this increase in the amount of synaptic protein inside of microglia in AD compared to controls. We were able to look then in culture because in the brain, the human brain, that's very important because we're seeing what happened in the actual disease, but it's a snapshot at the end, so we can't see what's the chicken or the egg, what's the cause or effect. So for that, we moved to model systems, and here we were very lucky to collaborate with Veronica Moron and some of our neurosurgeons here at the University of Edinburgh. And when our neurosurgeons go into someone's brain to remove a tumor or to remove a part of the brain that causes epilepsy, they have to take out a small amount of normal brain that usually gets thrown away. But recently, these kind patients and neurosurgeons have been donating that bit of normal brain to us, and we are able to isolate these little microglial cells and grow them in a dish over time. And in this experiment, we grew these microglial cells in a dish, and then we took synapses from human Alzheimer's disease brain or control brain and fed them to the microglia. And they were labeled with something that makes them glow red when they get eaten by microglia. So here I'm showing you human microglia in a dish. The red is showing you the synapses that they've eaten, and you can see them combined here. And when we look at how fast and how much these microglia eat synapses, what we find is that these microglia eat the human AD synapses more and faster than the control synapses. So the AD synapses are shown in red here, the quantification of the controls in black, and you can see there's a small effect, but there's significantly more area under these curves, so more synapses eaten from AD brain, and also the time that it takes to eat them is less in AD brain. So we were only able to do this now twice because of course it's very rare to get these valuable donations of surgically resected tissue. But we're also able to do this with mouse brain. And I can show you here a video of how this works in mouse brain. So we take mouse microglia, and you can see over time you get more and more red. So it starts out, they're labeled with blue in the nuclei, and you can see the cells in the phase contrast in the back. And I'll play the video again, and you see over time you get more and more red, which is the human synapses being eaten. You can see that here. So we're able to repeat this with lots of different mice, and we robustly see that mouse microglia also eat human Alzheimer's disease synapses more and faster than control synapses. And now a last little bit of data I'd like to share with you is I talked about genes already. I've talked about some of the proteins involved, but we also mentioned at the beginning that lifestyle changes can be increasing your risk for Alzheimer's disease. And one of our PhD students, Anna Stevenson, has done some work with her co-supervisor Riccardo Marioni and I, and she's found links between lifestyle and inflammation in the body and cognitive decline with age. So Anna has looked at people from the Lothian birth cohort group, so these are people who've had cognitive testing across their whole life, and she sees that markers of DNA changes caused by chronic systemic inflammation go up as people get older. And this marker is associated with smoking and high BMI, which is linking lifestyle to brain changes. This marker of DNA methylation of inflammation was also associated with poor cognitive skills during aging. And markers of aging on DNA are associated with more microglia in the brain. So this is showing you the increase over time of the inflammation marker in the blood. What we're showing you here is a marker of brain age. So a marker of age acceleration in brain is highest in the hippocampus, that part of the brain that dies in Alzheimer's disease. And I also saw those microglia, which I showed you just now, that eat synapses, also get higher in that same part of the brain that has the oldest epigenetic age. So this is a little bit of data now from our group linking lifestyle and inflammation with cognitive aging and microglia. So to summarize what I've shown you from our lab, I've shown you that genes, age, and lifestyle can contribute to protein accumulation. This, through interactions between neurons and microglia and astrocytes, or glia, is leading to synapse degeneration, which we think is really important for cognitive impairment. And in just the last minute or two, I'll tell you a little bit about where I think we're going with research in the future. So there is a drug treatment now in trials based in a small part on our research in the lab, and I'm on the scientific advisory board for this company now because of my work with them, so that's a potential conflict of interest. But I wanted to tell you about it because it's part of the story to show that research works. At a Society for Neuroscience meeting, which is a great big meeting, happens every year in the United States, in 2012, Dr. Catalano approached me to ask about helping their work because they think they had a drug that can take A-beta off of synapses. And we helped them. I won't go through these data, but we helped show that this drug can remove A-beta from Alzheimer's patient brain tissue that's been fresh frozen. And now they're trying to remove A-beta from synapses in human clinical trials, and we're helping them with this trial. So we don't know if it will work yet, but this is one way that research goes from the lab bench, in our case, to clinical trials. And I want to say that collaborations are really essential for science to move forward. Scientists go to networks like conferences like this. We want to promote rigorous and reproducible science, and we want to keep encouraging young people to join the scientific endeavor to solve this horrible problem of Alzheimer's disease and dementias. And it's also important that we engage the wider world and scientists explain to the public who funded our work why it's important, encourage policymakers to invest in research, and have a dialogue with society about the value of science, because it's very important that people understand that we're doing our best, and it's important to pressure your policymakers to create more research so that we can solve these diseases. So I'll just finish by saying, I hope I've convinced you today that our group uses multiple approaches to make progress in understanding the brain changes that cause Alzheimer's disease, and that future breakthroughs, I think, need collaboration between scientists like us and the wider society like you. So I hope that by looking through our looking glass, we'll be able to, in the future, develop the life-changing treatments we need for dementias. And I'll end with thanking my fantastic lab group, and a special thanks to our patient donors and their families, because without them, we wouldn't be able to do this research. So thank you very much for listening.
Da sier vi tusen hjertelig takk til Tara for en fin forelesning som gjør at vi nå vet litt mer om hva som foregår i hjernen på Alzheimer-pasienter. Og det gjør at vi klar for å stille eksperten vår spørsmål. Tara, can you hear me?
Yes, hello.
Wonderful. It's so nice to meet you, and thank you so much for a wonderful lecture. So are you ready for some questions from the audience?
Absolutely. Thank you for the invitation. This is very exciting, and for your wonderful introduction and videos.
Okay, Tara, the first question that you will receive today is the following. Can physical activity directly protect against Alzheimer's? And if it can, how is that possible?
That's a great question. So we can't say for sure that physical activity can directly prevent Alzheimer's yet, because the kind of data that we have is looking at lots of different people and asking whether people who exercise more have less chance of dementia. And that is true, and that's well established so far. What we don't know is whether the exercise is directly protecting the brain or whether there are other things about the lifestyles of people who exercise that are protective. But what I would say is exercise protects your brain likely through your vascular system. So keeping your heart healthy and keeping the blood flow to your brain healthy is very, very good for your brain structure. And that's how we think this exercise might be preventing Alzheimer's disease. But as I say, we have a long way to go yet to conclusively prove that.
Okay, wonderful, thank you. So next question to you is, do you know what it is in the synapses from Alzheimer's disease patients that make microglia to eat them faster than healthy synapses?
Another fantastic question. If we knew that, we could make $1 million and try and drug it. There are good ideas. So there's some data from mouse models that suggest that part of the innate immune system, the complement cascade, is very important for this. And that at least in mouse models of Alzheimer's disease, there's a little peptide or protein called C1q that is tagged on the synapses as an eat me signal. And then the microglia see the signal and they come and eat the synapses. It's less well established whether that's the key thing in human Alzheimer's disease brain, but we're looking into it, and so are many other groups, and I hope we can find that out, and that could be a point of intervention to maybe stop the synapses from being lost. But it's important to know we don't know whether the synapses are being eaten because they're dead and sick and need to be eaten and cleared, or they're being eaten when they're healthy and functioning. So we still, again, got a while to go before we fully understand this.
I see. Exciting times. Okay, so the next question is the following. In the very general terms, do you find results from your studies in different species like flies, mice, or human cells to converge towards the same result?
Yeah, that's another fantastic question. Thank you for that. Yes, that's why we use many different systems, because it's very difficult to be sure that what we see is going to be relevant to human disease if we see it in a fly, for example. But in raw terms, yes, we see some things that we find are very reproducible across different systems and across different cohorts of people. For example, when I did some work before I moved to Edinburgh, I was in Boston at Harvard Medical School, and we found data in people from Boston in the brain pieces they donated. And then we were able to see the same changes in brains that we got from people in Scotland, which is good to see, because if it's a widely applicable and robust biological phenomenon, it's more likely that if we target it, it will help the most people possible.
Okay. And this is a great question too. So why can't we just remove amyloid beta or tangles from the brain?
Yeah, great. So people have tried that, and removing amyloid beta from the brain when people already have Alzheimer's disease does not seem to be helpful. So you've seen from my work that I work in a lab. I'm not a medical doctor, but I follow the literature closely for clinical trials. And what happened is when people were vaccinated against A beta, either by giving them an active vaccine or by giving them antibodies directly to A beta, it successfully lowered levels of A beta in the brain, but the people didn't get better. In fact, some people got worse. And we think that might be because the stage at which this treatment was given was so far down the line that the brain was already full of A beta and tau. So the question about could we remove tau is a very good question, and the trials to try and do that are coming along. They're ongoing. There are immunotherapies that people are working on, and there are therapies with antisense oligonucleotides, so tiny bits of RNA, just like the new vaccines, the RNA vaccines for COVID, these tiny bits of RNA that go in and target the tau RNA and stop it from being made into tau protein. But those are at early stages, and we don't know the results yet. I think it's a promising strategy.
Okay, thank you. And so, how is it possible that social interaction may protect us from Alzheimer's disease?
Yeah, great question. And so there's something I should have explained more clearly in the lecture, and that's the studies about lifestyle, like social interaction and exercise. The data we get from them can't prove causation. So we see this association, but particularly in the social isolation data, it could either be that people who don't have interactions with others don't have as many synapses, because synapses are made and grown and get stronger with activity, and that contributes to their disease risk. Or it could be that people in the very early stages of the disease already have brain changes that mean that they're less likely to go on and socialize because they have problems talking or problems thinking or problems remembering. So we don't know the cause and effect, the sort of chicken and egg relationship yet, but my speculation is that there is probably some contribution of social interaction, just like there is with physical interaction, in building your brain's reserve, building your synapses and building strong connections that protect your brain from the pathology.
Okay, this is great. I have one last question for you, because that's all we have time for. And the question is, tau and amyloid beta, do these interact, or are they independent factors in the disease?
Another fantastic question. Well done to all of your audience. So the answer is we don't know fully. We know that by definition you have to have both in your brain to have Alzheimer's disease. And we think that amyloid beta is an early part of the disease that then kicks off a process that includes changes in tau that is very toxic. But how exactly that happens is one of the big outstanding questions in the field that we need to resolve. There are some good data from animals suggesting that amyloid kicks off sort of an inflammatory microglial and astrocytic process that then is damaging when tau is present. So there's lots of good hints there, but we don't know for sure the full story yet.
Okay, wonderful. Thank you so much for your willingness to participate today. We have learned a lot from you.
Thank you very much.
Okay. Da sier vi tusen hjertelig takk til Tara, men ikke minst til alle dere andre som har bidratt med helt fantastiske spørsmål. Og en helt viktig del av det å drive med forskning, det er å sørge for at dere er med på laget vårt. Og det gjør at jeg er veldig optimistisk til at dere er klar for foreleser nummer 2. Og neste foreleser, det er en som har jobbet innen Alzheimers-feltet i mer enn 30 år. Og det hun har fokusert på i det siste, det er det faktum at det viser seg at noen områder i hjernen ser ut til å være spesielt sårbare for Alzheimers sykdom. Og i tillegg til å fortelle oss litt om forskningen hun har gjort som fokuserer på nettopp den sårbarheten, så skal hun snakke om hvordan sykdommen sprer seg fra de sårbare områdene og til andre områder i hjernen. Den som skal snakke i dag er Karen Duff. Hun er professor ved University College London. Vær så god, Karen.
Hello, everybody. Thank you very much for inviting me to talk at the opening of the KG Jebsen-senter. As you can see from my first slide, I've relocated from America, where I was for 27 years, to take up the director position at the Dementia Research Institute at University College London. I'm sorry I can't be with you in Norway, but hopefully the situations will change fairly soon. So I want to talk to you today about two aspects of Alzheimer's disease that I think are very important to consider going forwards. And these two aspects are the phenomena of pathology spread, where disease-causing proteins accumulate in one area of the brain at the beginning of a disease, but spread through the brain as the disease progresses. The second phenomena is selective cellular vulnerability. This is where disease-causing proteins accumulate in particular cells, but spare adjacent, very similar-looking cells. This is the concept of cell vulnerability and cell resistance or resilience. Now, these two phenomena are, I think, going to be very important in how we diagnose a disease at the earliest stage possible. And currently, there's a very long buildup of these pathological proteins in the brain before dementia starts, which gives us a great window to explore for very early cognitive dysfunction. In addition, knowing the makeup of individual cells which are impacted by pathology will allow us to better target therapeutics that can specifically operate in those affected neurons, sparing the well-looking cells around them. So the idea of pathology spread came from post-mortem mapping studies performed by neuropathologists such as the BRACs. And here is an example showing the distribution of amyloid plaques on the top and the distribution of neurofibrillary tangles on the bottom. And you can see the amyloid plaques are fairly widespread in the brain at all stages of disease, a higher concentration in the frontal cortex at the beginning of the disease. But the distribution of neurofibrillary tangles looks quite different. There's a high concentration of neurofibrillary tangles in the entorhinal cortex at the very earliest stages of the disease, which spreads through the limbic regions into the isocortical regions, with dementia starting as pathology starts to reach the neocortical areas of the brain. And this stereotypical and predictable pattern of tau distribution in the brain in Alzheimer's disease suggested to pathologists such as BRAC that perhaps the connections between the neuroanatomical connected regions was important for the phenomena of pathology spread. Perhaps the pathology was spreading across synapses that link these areas together. Now, we were very interested in modeling this initial pathology development and the spread of the pathology in a mouse model. And we created a mouse model called the EC-tau transgenic mouse through crossing two types of mice together, one which expressed the tau protein under the neuropsin promoter, and the other which expressed a pathological form of tau, which would give rise to the well-defined tangles that you would see down in the bottom here. So when we looked at these mice at a young age, before 18 months of age, we saw the distribution of tau here in brown, human tau, in the medial and lateral entorhinal cortex, as expected. That's where it's expressed, but also at the ends of the axons, the axon terminals that terminate here in the middle molecular layer. Now, this is a sort of normal distribution of the protein, mainly in the axons, a little bit accumulating in the cell bodies. When we looked at the mice when they were older, over 20, 24 months of age, right up to 36, 40 months of age, we saw a very different distribution of the protein. Tau protein was now accumulating in the cell bodies. It was thioflavin S positive in regions of the entorhinal cortex, suggesting it was mature tangles with beta sheet arranged tau. But it was also now spread out in neurons that were outside of this area where human tau was expressed. And we were able to see tau accumulating in pyramidal, neurons outside in the perirhinal cortex and up into many of the other cortical areas. It accumulated strongly in the pyramidal layer of the CA1 fields, and it accumulated in granule cells, cell bodies, here in the dentate gyrus. Now, the distribution of tau in these areas was very reminiscent of what you would see in early Alzheimer's disease. The areas that were impacted by tau pathology in these earliest stages are very interesting because they house the cells involved in spatial navigation, spatial orientation and navigation. These are the entorhinal cortex, medial entorhinal cortex grid cells, the hippocampal place cells, and the whole slew of other specialized cells involved in navigation. The region that's impacted in humans, at the very earliest stages, the transentorhinal and medial entorhinal cortex regions, is probably giving rise to some of the deficits in spatial orientation and infamiliarity in places that patients with the earliest stages of Alzheimer's disease suffer from. We wanted to look at the impact of tau pathology here in our EC-tau mouse using in vivo recordings, and we looked at, because the impact on the medial entorhinal cortex, we looked at grid cells in the medial entorhinal cortex of these mice, and we were able to show that with age and as the pathology worsened from 14 to 30 months, the grid cells became destabilized. We also showed that this was associated with progressive cognitive impairment between 14 and 30 months of age as the pathology worsened and spread. And there was difficult deficits in spatial navigation as evidenced by performance in the T-maze. In addition to cognitive impairment, there was neurodegeneration. And this is an MRI-based assessment of atrophy in these EC-tau mice at 14 and 30 months of age. And you can see from this hotspot that the area of atrophy in these mice is co-located with the entorhinal cortex and spreads out into more areas of the entorhinal cortex and into the hippocampus as the mice develop worsening pathology and it spreads through the brain. Interestingly, the atrophy identified by MRI did not correlate with overt cell loss in the hippocampus. It precedes it. So we think that this is probably loss of synapses. So one of the things we're very interested in doing is identifying cognitive dysfunction in the EC hippocampal circuit as early as possible in the prodromal phase, when we currently can't see any dysfunction, but we know that pathology is starting. And we've been very interested in applying novel behavior tests. And here you see a mouse running a virtual reality setup with indwelling recordings to monitor the brain activity. And we think that this can be paralleled with virtual reality based navigation tasks that can be performed on humans. So we and several other groups around the world are very interested in applying these novel tests based on virtual reality, where we can correlate mouse behavior with human behavior as much as possible and use the mouse to explore the basis of this dysfunction. So coming back to the pathology, as I mentioned, is quite revealing about how the pathology is spreading from cell to cell. And one of the things that's particularly revealing is the idea that the tau can spread from the entorhinal cortex through the middle molecular layer into the granule cell layer of the dentate gyrus across a single synapse. This synapse occurs in the middle molecular layer, and we can use that synapse to think more about how tau is crossing from one neuron to the other. Now, because the mice were designed so that the expression of tau in that dentate gyrus granule layer was almost negligible, almost nothing, because we could see tau pathology developing in the granule cells, we were able to make statements that tau was able to leave the neurons that it was expressed in from the entorhinal cortex, cross the synapse, and be taken up by dendrites coming from the granule cells, where it would then accumulate. This idea that tau could actually leave one cell and accumulate in another was completely novel. The behavior of tau normally is that it's intracellular, it's bound to microtubules or in the cytoplasm. It is not meant to be extracellular. So just by the design of these mice, we were actually able to make some quite significant statements about this new behavior for the tau protein. We looked by immuno-EM at the location of tau in that middle molecular layer of the mice. And here we could clearly see tau, which was labeled with an antibody MC1 and the gold beads for immuno-EM. We could see it clearly accumulating in the postsynaptic compartment of this dendrite. And we could see it clearly crossing the synaptic cleft from the presynaptic to the postsynaptic compartment in this region. We could also see tau accumulated on synaptic vesicles.
Hello, can you hear me?
Where you see it decorating the outside of the synaptic
Can you hear me?
vesicles in the presynaptic compartment. And we could also see it present in some vesicles that were not synaptic vesicles, also within this area. So here it is in a number of different morphology vesicles within the middle molecular layer. Now, we were very interested in whether the association of tau with these vesicles actually gave some insight into how it was propagating between neurons. And we were asked the question, how could tau from cell A, shown here, the presynaptic compartment, cross into cell B, shown here as the postsynaptic compartment? How could tau move across the synapse? There are a couple of ways that have been implicated. One is that tau could be free, released by a degenerating presynaptic compartment, released into the synaptic cleft and taken up by the postsynaptic compartment, either through the membrane, invaginated through a vesicle, or picked up by a receptor. Or tau could move through vesicles that were secreted. And this idea would be that tau was budded off from the presynaptic compartment, transported across the cleft, and endocytosed into the postsynaptic compartment, where it would then misfold and form more tau aggregates. We were curious to follow this and look further whether this could be the case. And to do this, we took human brain, Alzheimer's brain tissue, and we gently dissociated the tissue and released the extracellular vesicles, which are a heterogeneous collection of vesicles that are outside of the cells. When we looked at tau within those extracellular vesicles, we saw that the paired helical filament structure was recapitulated in tau in these vesicles. And even more surprisingly, the tau within these vesicles took this C-shaped architecture that had been seen in tau from the whole brain by cryo-EM specialists such as Anthony Fitzpatrick and Ben Falcon. And this was a remarkable finding, that the tau within the extracellular vesicles is actually the same form, has the same conformation and structure as the tau from the whole brain. One of the things we're curious about is whether targeting tau in the postsynaptic compartment as it's spread from one neuron to another might be a good therapeutic target to prevent the further spreading of the pathology through the brain. We had been looking at the ability to activate the proteasome through phosphorylation. And we had published a paper in 2016 showing that cyclic AMP mediated phosphorylation of proteasome could increase the ability to degrade tau and ameliorate cognitive impairment in various mouse models. We wanted to make this more specific though, and target this event, this cyclic AMP mediated enhanced activity of the proteasome, just in the postsynaptic compartment. To do that, we used this G-coupled protein receptor, GPCR, PAC1, which binds the ligand PACAP and activates PKA and the phosphorylation of the proteasome, but only in the postsynaptic compartment. When we did this with PACAP, we could see enhanced tau clearance and amelioration of cognitive deficits in the mouse models. We've published this both on bioRxiv, and it's currently in re-review at Science Translational Medicine. Now, we don't think that PAC1 is a therapeutic, but we wanted to sort of show that by targeting specific compartments that you know where your pathological protein has accumulated, you might be able to more specifically target your disease-causing proteins in a way that has less side effects possibly than targeting these cell systems throughout the brain. So I now wanted to switch to the second part of the talk, which is why do tangles develop in some neurons but not in others? And here you can see in this immunohistochemistry representation from the entorhinal cortex in early AD or in late AD, the blue cell is stained with a histological dye and has no tangle, but cells right next to it, neurons right next to it, pyramidal neurons, have accumulated tau shown in brown. And this is maintained both in early AD and in late AD, this selective vulnerability of particular neurons. Now we've looked, various people have published how selective vulnerability manifests in Alzheimer's disease, and several studies have shown that excitatory neurons throughout the brain are particularly vulnerable to tau pathology, whereas inhibitory neurons seem to be spared. And various cell types in different regions seem to be vulnerable, whereas other cell types seem to be spared. So we've been very interested in following up on this selective vulnerability idea and wondering, what is it about the vulnerable neurons that makes them vulnerable? Are there differences in proteostasis between a vulnerable and an invulnerable neuron? Metabolic activity and firing rate, endo or exocytosis and intracellular trafficking? Is it the morphology of the neuron itself that lends itself to be more vulnerable? For example, long axoned neurons are perhaps more vulnerable because of the length of the axons. Calcium homeostasis has been implicated, and cellular stress differences. And this is really what we wanted to get at, was what was different between what we knew to be a vulnerable neuron and what we knew to be an invulnerable or resistant neuron. We performed a couple of studies. Here is one of the published studies where we were looking at proteostasis as a differential expressed phenomena. And here we just used published RNA-seq data sets, looked at the difference in proteostasis-related pathways between excitatory neurons and inhibitory neurons in a vulnerable area. And doing a co-expression network hub analysis, we came up with a number of genes and pathways that looked like they were differentially expressed. And we're following up with one of these, the BAG3 autophagy promoting pathway, which looked like it was higher in inhibitory neurons than in excitatory neurons. However, we want to go on now and really get down to single cell level, not looking at data sets that are published, but generating our own data. And a number of groups are doing this. And there are a number of ways that you can approach this issue of looking at selective vulnerability. We're very keen to look in situ, in the tissue. And we've been started by doing a couple of these approaches listed here, possible approaches. One is laser capture microscopy, where we can pick out immunohistochemistry stained neurons within the entorhinal cortex, layer 2 of the entorhinal cortex, and we're doing both transcriptomics and proteomics off of those. But we're also using approaches such as the Visium, where you can take a spot you can sequence a spot of an Alzheimer brain or a mouse model brain and generate RNA profiles from areas that have pathology versus do not have pathology. More sophisticated techniques such as in situ sequencing are in development. We have an active and ongoing interest in these techniques. I lastly wanted to finish off by just making the statement that these ideas of pathology spread and selective vulnerability are not unique to Alzheimer's disease. They are seen in every neurodegenerative disease. Here's Parkinson's, showing the progressive spread of synuclein, ALS, progressive spread of TDP43. And here's the selective vulnerability idea, showing the areas that are impacted early on in each of the diseases, showing that it's different cell populations, different regions. So I think understanding these concepts of pathology spread and selective vulnerability are applicable to a very wide range of neurodegenerative diseases. And I want to just finish off with thinking about a couple of key questions. The first is, we want to ideally target drugs that will be effective in patients at the earliest stages. So should we take into account the molecular makeup of different cell populations that are vulnerable when we're designing our drugs? I think so. I think that's the basis of our work, has shown the differences between the cell populations we need to take into account. And how better can we identify common but druggable pathways that are effective in these vulnerable cell types? I like looking at hub analysis when you're doing RNA-seq analysis, because it really homes in on the key druggable pathways amongst all the many pathways that you might be tempted to target with a drug. And very importantly, how can we identify dysfunction in the entorhinal cortex hippocampal circuit at a much earlier stage, during what is currently the prodromal or preclinical stage, where you cannot diagnose the disease through current behavioral tests. This will allow us to administer drugs earlier, and which will probably have a greater impact on their efficacy. So I want to just thank my lab, my current lab at UCL, where I have now 4 or 5 postdocs and a couple of PhD students, my lab at Columbia, which I still run partly. I still have a position at Columbia, many people who have contributed to some of the work that I've shown and the ideas behind them, and my many collaborators around the world. And thank you very much to everybody for listening. And I hope there's some questions.
Da sier vi tusen hjertelig takk til Karen for en grundig innføring i hvordan genteknologi og bruk av musemodeller kan gi oss innsikt i sykdomsprosessen tilhørende Alzheimers sykdom. Og i tillegg så det sånn at vi ser at disse studiene også kan inspirere til utvikling av adferdstester som kan brukes til både mennesker og til mus, og på den måten minske avstanden mellom studiene fra ulike arter. Og så det liten tvil da at siden jeg selv jobber med mus, så jeg ekstra begeistret for hva det Karen hadde å si. Men i dag så det altså dere som setter agendaen for programmet vårt, og vi skal gå over til å stille Karen spørsmål som dere syns mest interessant. Karen, can you hear me?
Yes, I can hear you.
Wonderful. So I'm Emilia, and I'm, as you know, the anchor for today, and will be conveying questions for you from the audience. And are you ready for questions?
Yes.
Wonderful. Okay, so the first question that you will receive today is the following. If Alzheimer's pathology can spread within the brain sort of like a virus, can one get infected from others with Alzheimer's disease?
That's a question that has been addressed a couple of times, and the answer is still a little complex. So some studies have tried to see whether if you ingest or take in an infected tissue, whether you can get the disease. And some of those experiments have been done in people who received human cadaver-derived pituitary gland extract to correct their height problems. And later on in life, it was looked at whether those people went on to have a higher incidence of Alzheimer's disease. Now, some studies said no, they didn't, and some studies said yes, they did. So there's clearly a potential for ingesting tissue that is infected with Alzheimer's disease to cause Alzheimer's disease in a person who takes it in. And this is a little analogous to these prion diseases like mad cow disease, and diseases that's caused by a protein spreading through the body. But I think for most people, and this is a very important point to note, to realize, for most people there is no risk, I think, of Alzheimer's disease spreading through surgery, through your grandmother sneezing on you. There's not going to be a risk to most people from an infectious Alzheimer's agent. The experiments that were done were really very special to really test the system at a very extreme level. So no, I think it's not a risk that we should be worried about.
Okay, thank you for clarifying that. Okay, so the next one, does Alzheimer's disease occur naturally in mice and rats? And if not, why is that?
Yes, Alzheimer's disease is pretty much a uniquely human disease. And you do get age-related accumulation of some of the tau pathology. You get some of the amyloid accumulating in some of the animals, not rats and mice. They don't live long enough to get it. But if you look at monkeys, polar bears, some of the larger, longer-lived animals, you do see some of the pathology. But it never really evolved into what we would consider to be Alzheimer's disease. So it may be they don't live long enough. It may be that their brain metabolizes these proteins at a much higher rate and they don't accumulate to much of a level. But no, you really don't get Alzheimer's disease in most other animals. Some primates have some symptoms that we think look Alzheimer-like, but it's not the full-blown disease.
Wonderful. And following up on that, what is the point then of doing all these studies in mice? And mice are very different from humans. So what is exactly the relevance here?
Yeah. So obviously it's pragmatic, meaning we need to do studies in an animal model. We have to be able to manipulate the systems. We have to have a system where we can test drug candidates. Mice and rats are very convenient model systems. We have improved them hugely by introducing these genes that in humans cause Alzheimer's disease. So this would be amyloid causing APP or the tau transgene. If you put those human genes into the mouse or rat, you do get a reasonable similar disease forming that we can then use to test drugs and to look at mechanisms. Now, the mouse and rat models have not been great in the past. They're getting better every year. They look much more like human Alzheimer's disease as the years go on. And I think that the next generation of these animal models, mouse models, will be much better, much more predictive of what will happen in a human population when you give them a drug or something.
Okay. And so, next question is, can a bad sense of direction lead to Alzheimer's disease? For example, if the grid cells and the place cells are dysfunctional?
It doesn't go that way, it goes the other way. So you really have to have the pathology to cause the dysfunction of the grid cells and your ability to navigate. I don't think having a bad sense of direction gives you Alzheimer's disease. I hope not. I'm particularly bad at navigating through buildings with lots of different exits. But I think, no, it doesn't work that way. I should say that activity in the brain, where neurons are firing faster than normal, could precipitate Alzheimer's disease. We know neural activity can actually cause some of these proteins to accumulate faster. But that's not the same as having dysfunction in a grid cell field.
Okay, and so the next one, can you have Alzheimer's disease without any amyloid plaques?
So we get ourselves into a bit of a tangle here, a bit of a mess, because Alzheimer's disease has been defined by its pathology. So in theory, if you don't have amyloid plaques and tau tangles, you don't have Alzheimer's disease. But what is Alzheimer's disease? It's also it's a type of cognitive impairment, which is due to dysfunction in the entorhinal cortex hippocampal circuit and cortical areas. So if you have that type of memory impairment, do you have Alzheimer's disease? So I think because we are calling Alzheimer's disease plaque and tangle disease, it's not officially classed as Alzheimer's if you don't have amyloid or you don't have tau. However, these definitions are being revised. There are now considered to be subtypes of Alzheimer's disease that perhaps don't have amyloid or don't have tangles in the way that we would normally define them. This is all under review, and there's a lot of discussion in the field as to how we should classify Alzheimer's disease beyond just having plaques and tangles.
Okay, but somewhat related to that, where do these disease-causing proteins come from in the first place? Do we know that?
So the disease-causing proteins are all naturally occurring in the brain. They are part of normal pathways that are very important to brain function. What happens to make them go wrong and give you Alzheimer's disease, making them accumulate and aggregate and kill neurons, is still not quite clear. However, the greatest risk factor for Alzheimer's disease is aging. So aging seems to set off some events in the brain where these normal proteins start to behave abnormally. A small number of people have mutations in these genes and others, lots of different genes implicated now. But these are a small population of the patients who have Alzheimer's disease. They're usually referred to as familial Alzheimer's disease or the APOE Alzheimer's disease. So by and large, we don't know what sets it off. And that's a really big question. We're starting to realize that Alzheimer's disease probably starts in the brain about 15 years before you have symptoms. So everything we've done looking at the brain at the time when the first symptoms appear is probably not going to be informative for why these proteins went wrong in the first place. We're going to have to look in this prodromal, very early stage of the disease, which at the moment we can't identify people who are in that stage. We need better cognitive tests. So no, we don't know what sets it off, but aging is clearly a major risk factor.
Okay, wonderful. Thank you so much again for coming here today and for answering all of these questions. Thank you so much.
Thank you.
Da sier vi tusen hjertelig takk til Karen, og ikke minst til dere som har bidratt med å sende inn en hel horde med gode spørsmål. Det er veldig gøy å se at engasjementet strekker seg utover bare her hos oss på senteret. Det vi skal videre til nå er neste foreleser, og den eksperten heter Emrah Düssel. Han er nevrolog og professor ved Otto von Guericke-universitetet i Magdeburg. Og til oss så skal han fortelle at han henter informasjon både fra bildediagnostikk, men også fra væsken som omslutter sentralnervesystemet vårt, for å kunne si noe om hvordan sykdomsprosessene i Alzheimers kan faktisk påvirke hjernens funksjon. Og igjen så minner jeg om at dere er helt nødt til å bruke Slido og engasjere dere i å stille spørsmål til foreleseren vår. Da sier vi bare vær så god til Emrah.
Ladies and gentlemen, dear colleagues from the new Jebsen-senter for Alzheimer's disease, I'm really honored that I can speak at the opening symposium of your new center. And I'm also really thrilled that you, as leading cognitive neuroscientists in the world, are now joining the fight against Alzheimer's disease. I very much look forward to the innovation that will come from your sides, to the impact that you will have on the field, and hopefully to our future collaborations. So I'm going to talk about our work at the German Center for Neurodegenerative Diseases, particularly about the effect of Alzheimer's pathology on brain function. So the German Center for Neurodegenerative Diseases is a government institution that spans across 10 university-associated sites in Germany and really spans the whole translational research spectrum in Alzheimer's disease. And we are located in Magdeburg. And our concern in Magdeburg is particularly cognitive neuroscience, imaging, and plasticity-related interventions. Well, let's dive right into Alzheimer's disease. We have two major culprits. One is tau that starts in memory regions, the medial temporal regions in the brain, and then from there spreads to other areas really across the whole brain. But initially being really limited to these memory regions. And then we have amyloid that is much more widespread initially, basically affecting most of the cortical mantle. And as you can see from these numbers in blue, these early stages of tau pathology where there is only limited pathology in the brain that is visible here as black staining is actually quite common in individuals who are cognitively normal and don't complain of memory dysfunction, so 60 to 80%. Amyloid pathology is also not uncommon, so 20 to 30% of older, seemingly cognitively normal individuals have amyloid pathology in their brain. So what we are trying to understand is how neurodegeneration, amyloid, and tau pathology impact on brain function and on cognition. In order to explain our work, let me look a little bit more closely into what all this means. So let's imagine we are looking at a piece of brain and then an area in the cortex. So in that region, of course, we have millions of neurons, and the tau pathology is basically in the neurons, intraneuronally, and the amyloid pathology, particularly the plaques, are in the extracellular space. Now we can measure these pathologies through different ways. So one is by looking at their presence in the cerebrospinal fluid that we can access through a spinal tap. Or we can conduct molecular imaging, such as positron emission tomography, and then look at amyloid and tau depositions by using radioactive agents that bind to them. We can, of course, also look at neurodegeneration by measuring the volume of brain tissue. And the idea there is that the rich and complex layer organization of the brain at some time in the Alzheimer's disease process starts to suffer from amyloid and tau pathology, and we have then loss of synapses and neurons, and the consequences that we have shrinkage of the cortical volume, so volume loss, and that's a measure of neurodegeneration. So we have amyloid and tau pathology, we can measure neurodegeneration, and the question that we really want to address is how does this impact on brain function? So in our studies, we obtain cerebrospinal fluid through spinal taps. We conduct brain measures using MR, but also we measure brain activation using fMRI, so functional magnetic resonance imaging, and then we bring all this together in order to understand the relationship between brain function and pathology. And in doing so, we consider different kinds of memory processes, different kinds of memory, different types of memory representations. So it's a very rich field of cognitive neuroscience that we then apply in order to understand basically how these pathologies impact on brain function and cognition. The one very large and important study that we conduct in that regard in the German Center for Neurodegenerative Diseases is the so-called DELCODE study, the Longitudinal Cognitive Impairment in Dementia study. This is a multi-center study across the 10 sites of the DZNE. And really we recruit over 1,000 individuals into this study that come to our clinics once a year. Some already have follow-up data over 5 years, and the range is from cognitively unimpaired to cognitively slightly impaired to early Alzheimer's disease. And here are the numbers. We have 200 healthy control subjects who are cognitively unimpaired and don't have any memory complaints. We have first-degree relatives of Alzheimer's patients. Then we have a very special group that I will talk a bit more in detail about. We have 400 subjective memory complainers. So these are older adults who go to a GP and seek advice because they believe that their memory is deteriorating. But on our neuropsychological testing, they perform normal. And so they don't fulfill the criterion for being cognitively impaired, and therefore are classified as subjective complainers. But then of those complainers, some actually are cognitively impaired, mildly impaired in our neuropsychological tests, and those we call mild cognitive impairment or MCI, and of those we have 200 individuals, we have 100 Alzheimer's patients, and those, as I said, come for regular follow-ups. So how do we measure brain activity using cognitive neuroscience approaches? So one way is we look at different memory processes using tasks that participants do while they lie in the scanner and while we measure their brain activity. This is one of the tasks that we use. It's basically a photographic recognition memory task. So in the scanner, participants see images like this, one after the other. Every few seconds a new photograph, and they have to make a very simple judgment whether it's an indoor or outdoor scene that's displayed. And we use a trick. We repeat some of the photographs over and over again so that the brain of the participants gets used to that photograph. And that allows us to look at how the brain responds to the novel stimuli, so the activation induced by the novel ones as opposed to the repeated ones. So it gives us the so-called brain novelty response to a new novel information. So that's a measure of encoding information into the brain. And then this whole thing takes only 8 minutes, but once participants leave the MR scan, 70 minutes later, we actually test their memory. So we make a recognition memory test by showing them photographs they've seen in the scanner and new photographs, and they have to tell us which one is new, which one is old. And that gives us a measure of their memory performance. So this is, for instance, the data that we have in the DELCODE sample across the 10 sites. As you can see, we have fMRI data from 55 cognitively normals, 55 relatives of Alzheimer's patients, 163 cognitively normal, non-complaining older adults, then 221 subjective memory complainers, and 81 mild cognitively impaired patients. You can see their years of education is roughly matched. And so how do the data from such a study look like? So here you see the memory performance. This is basically the bars show you the recognition memory accuracy, so a higher bar is better. You can see that healthy controls and subjective complainers perform roughly similarly, but once you have mild cognitive impairment, you are significantly impaired. And how does this relate? How does this performance relate to Alzheimer's biomarkers, so to amyloid and tau? So, well, there is no relationship to amyloid, but there is a relationship to the CSF, so spinal tap-derived measure of tau concentration. And you can see that the higher the level of tau is, the lower recognition memory accuracy. So recognition memory accuracy goes down as your tau levels in the CSF increase. So this shows us that tau apparently has an impact on cognitive performance. But how about function? So we have the novelty response, as I just mentioned, in the brain when participants see those novel images. And now we can look at how tau actually affects the novelty response. Now the area of interest is here. This is the hippocampus, a very important memory region. And so what we are looking at here now is where novelty correlates with tau levels in the CSF. And we have done this separately for those individuals whose amyloid levels in the CSF is normal and those who have pathological amyloid levels. And what you can see is that if you are CSF normal, there is no correlation of novelty with the tau levels, so there is no relationship. But if your amyloid levels are already pathological, then there is the quite clear case that your novelty response decreases with increasing tau pathology. So this is a negative correlation. So the more tau you have, the less responsive your hippocampus is to novelty. So apparently, in addition to the behavioral effect you have just seen, tau also reduces the way your memory reacts to novel stimuli. Now the question is, does this depend on neurodegeneration, so on volume loss, or is it a more direct effect of tau? Well, the answer is it does not depend on volume loss, because if we consider hippocampal volume, it doesn't really matter. The correlation remains as strong. So volume has no effect on this correlation, which shows that there must be a direct effect of tau, which reduces encoding of novel information, and that's not due to volume loss or neurodegeneration. Now, why is that important? It's important because it shows you that there may be a potential benefit of a tau-related treatment on brain function. And the reason is you can recover brain function, but it's hard to recover if you have already lost a lot of neurons. But if tau has an impact on function and cognition, even before you're losing neurons, then it's a good entry point for a treatment. And that's what you can find out with these types of cognitive neuroscience brain activation studies. And that's why I think they are very important. Now, what about the effect of amyloid? So apparently we have the correlation in the group that's amyloid positive. Now, this raises two possibilities. One is, if you have amyloid, this impairs brain reserve to cope with tau pathology. So maybe there is some way to cope with the toxic effect of tau, and amyloid impairs that reserve ability. The other possibility is that amyloid just makes tau more toxic where it acts. And we haven't really figured out which one of those is true yet. But they are both obviously important considerations and alternatives. Now, we can do something else in this study. We can look at the whole brain pattern and consider something that we call youthfulness. This is because we know how young adults activate their brain in response to these novel photographs. Now we can take basically this activation pattern and compare it with a statistical matching to the activation pattern of older adults individually. So for each individual, we can say how similar is that older person's brain activation pattern to the canonical pattern that we know young individuals show. And then we can basically plot here a similarity index, where highly similar would mean that you have a very youthful brain in terms of brain activation, and low similarity means that your brain shows some aging effect. So it's different from what a young person shows. Now it turns out that when we look at the subjective complainers, this measure actually shows quite a robust relationship to amyloid pathology. So what you see here is a similarity score, and here you see the CSF levels of amyloid pathology. And we see that more pathology means less similarity to young adults. And this is basically with different measures of amyloid and of amyloid pathology. Now, interestingly, we don't see this relationship in cognitively normal and non-complaining older adults. So we see that relationship only if you have memory complaints. So what does that mean? Now, why has amyloid pathology such a linear impact on the network or function pattern or youthfulness in the complainers, but not in non-complaining healthy older adults? Well, it looks like brain function is kind of unprotected against the increasing amyloid pathology in the subjective complainers, but there seems to be something that protects it in the non-complaining individuals, and so something that may act as a brain reserve. So what could that be? Now, it turns out that if we look at the volume of these medial temporal lobe regions, so of the hippocampus, and particularly of the subfields, so the hippocampus is a very interestingly organized, very mechanistic and rich structure with different subfields. And when we look at the volume of these subfields, we see something very interesting. So here in blue, you see the volumes of healthy controls, and in red you see the volumes in subjective complainers, and these here are those who are amyloid negative, and these ones are amyloid positive. And what's plotted here is the volume from this blue region, the dentate gyrus. And you can see that, and this is really surprising, that healthy, non-complaining older adults have actually a larger dentate gyrus when they are amyloid positive. Those who have subjective complaints, they actually have quite a substantial volume decrease here. And this is even more prominent when you look at another subfield, which is yellow here, the CA3 subfield. There you can see that actually the amyloid positive healthy controls have larger hippocampi than the ones who don't have pathology. Now you could say, this is really strange. Why should a healthy control person who has pathology have a bigger, hippocampus, bigger memory region than someone who doesn't have pathology? And I think the answer is that only those who have bigger hippocampi can be cognitively normal with amyloid positivity. So that's basically having brain reserve. And obviously this brain reserve is lost or not there to that degree in those who are subjectively complaining. Now, in addition to these types of interesting relationships, with new cognitive neuroscience methods, we can also find subtle cognitive deficits that are not apparent in normal neuropsychological tests. For instance, we see that subjective complainers have a very limited and specific deficit in memorizing spaces or rooms as opposed to memorizing objects. Now, what does this all mean? So I think it shows us that when we consider brain reserve and when we consider individuals who have increased brain reserve, then you can remain non-complaining despite the fact that you have amyloid pathology. But if you don't have brain reserve, you start having subjective complaints, a network change in your brain activity with increasing amyloid pathology and also subtle deficits. And if tau comes into play, you have marked hippocampal dysfunction and cognitive impairment. So that's a current working model that we have. Now, what we need is basically a way to translate these types of cognitive neuroscience aspects into the wider population and into healthcare. And here, digital health can be very important. So we are investigating whether smartphones can help improving cognitive assessments by forming that type of bridge from science to care. And so in our studies, we have the opportunity to compare the assessment by neuropsychologists with the assessment done on a remotely done on a smartphone when patients take the tests home and do it themselves. So we have, these tests really informed by cognitive neuroscience that people can do at home. And we already can show, for instance, these are data from the Delcode study, that what you perform at home has a very strong relationship to certain scores that are very important in the Alzheimer's field. And this tells us that it is actually feasible to use digital technologies for assessment remotely and in unsupervised ways. And I think this is really the future when it comes to implementing new therapies, for instance, antibody therapies that remove amyloid from the brain and which may be approved by the Food and Drug Administration maybe in the near future. And digital technologies basically allow us a situation where doctors prescribe the digital assessment, which participants or patients do at home, and then get a reliable report whether a person is cognitively impaired or not. So to conclude, modern imaging tools and advances in cognitive neuroscience really allow us to understand the impact of Alzheimer's disease on brain function. We can also identify mechanisms of brain reserve helping to cope with pathology. And digital technologies provide a rapid translation and dissemination with wide reach and impact for healthcare. I thank you very much for your attention, and thank you again for making me part of this ceremony.
Tusen hjertelig takk til Emra, som viste oss at det ser ut til å være en direkte link mellom noen av de spesifikke sykdomsforandringene ved Alzheimers sykdom og hjernens funksjon. Og som Emra understreker, så betyr jo dette at intervensjoner rettet mot nettopp de sykdomsforandringene kan gjøre det mulig å potensielt reversere eller tilbakevise de hjerneforandringene som gir svikt. Og det synes jeg er utrolig spennende å kunne si. Så med den optimismen der, så tenker jeg vi bare beveger oss videre til spørsmål direkte til Emra fra dere. Emra, can you hear me?
Yes, hello, I hear you very well.
Hey, nice to meet you. So,
Nice to
are you
meet you.
ready for
Thank you for
some
having me.
questions? Yeah, okay, wonderful. So, the first question that we have for you today is, can functional imaging during memory tasks be used as an individual biomarker for early Alzheimer's disease? And what are the potential challenges?
Yes, in principle, I think this is feasible. We have to conduct MR scans anyway, so that's part of the Alzheimer's assessment. Structural imaging with MR is the cornerstone of the clinical diagnostic workup in Alzheimer's disease, and I think it's not too difficult to implement in such a clinical setting in a specialized memory clinic. A standardized task to assess brain function as well. The problem is that the way to or the procedures that are required to bring such a procedure into a clinical standard are quite laborsome, so you need a lot of standardization, replication. You need very clear quantification and cutoffs. We are working on this, but I assume it will still be a few years before we can see something that's being used in a routine setting. Currently, fMRI is mostly used for research. And I think in research it plays an important role. Maybe, closer than using it in clinical routine is the possibility to use it in pharmacological trials.
Okay, thank you. And so next, is it realistic to expect elderly people with poor technological skills to perform a digital assessment at home by themselves?
Yeah, I mean, I think the interesting aspect of Alzheimer's disease is that, and you have also heard this from the previous speakers, is that the pathology is present before, clinically impairing symptoms emerge already 10 to 15 years. And in this long preclinical prodromal phase of the disease, people are, still capable of using their smartphones and interact with technology. And at least in our experience, this was not really a problem. Of course, once you move towards the clinical stage of dementia, things change, and then there is a very strong limitation to usability. But particularly when it comes to early assessment, detecting the early cognitive trajectory in Alzheimer's disease, smartphones are suitable. And, we see an increasing number of older adults that are using smartphones. The field, the usage is increasing quite rapidly, and probably in 5 years will be much more than today already.
And what about the type of imaging that you do? Is it possible that this type of imaging help with the treatment as well? Can you treat a damaged area specifically if you can see it in your MRI machine?
That's an interesting question. So I think the question is whether if you see that a particular brain area is dysfunctional, whether you can have an intervention that targets particularly that specific brain area. And I guess, that intervention could be a specific type of cognitive training that somehow allows this brain region to utilize its remaining resources, or maybe to reconnect to other brain areas so that it can be integrated again into a network. You could also think about, and obviously this is much more in the future, of stimulating brain areas. There are, methods of deep brain stimulation for certain brain regions, but it's not a common procedure that can be applied to many different areas. I think some labs are thinking about certain brain areas, such as the entorhinal cortex, as being a target for direct brain stimulation interventions. In principle, this is something where the field really is looking, I think, quite closely into what's possible beyond pharmacological interventions. This is also work in progress, and obviously centers like the one, the Jebsen-senter for Alzheimer's disease, can make important contributions to this field as well.
Yeah, thank you. We are, of course, happy to hear that. And so to the next, how are memory tasks versus navigation tasks affected by Alzheimer's disease? Does fMRI show the same thing in those tasks in patients with Alzheimer's disease?
Yeah, that's a very important question. I think spatial navigation is obviously a clinically very important question, because at some point this is a important clinical feature of Alzheimer's disease. in prodromal Alzheimer's disease, when we talk about mild cognitive impairment, it's already something that's measurable and evident, but it may be measurable and evident much earlier. So we're waiting for your findings there. The truth is that this is still in a young field. So, a well-informed spatial navigation task in Alzheimer's disease, in the research of this disease, there's not so much around. Looking at memory, the types of memory functions that I've mentioned has been around for a much longer period of time. So we still have to see and wait how these two things relate to each other. Obviously, there is a similar network that underpins both spatial navigation and the types of memory tasks that I've just mentioned. But how they directly mechanistically link together and how they are then affected by the Alzheimer's disease, I think that's something that will emerge over the next few years, hopefully.
Wonderful. And so during the lectures today, our viewers have heard dentate gyres mentioned several times today. And why is this region so important?
Yeah, it is a fascinating brain area to begin with. So people look at it, I think, quite often because it's a brain region that, one of the few regions that shows the potential for neurogenesis, that is creating new neurons into very late adulthood. So there is this potential for plasticity, for ongoing plasticity, and then it serves probably an important function in memory, namely the ability to separate in memory very similar events so that you don't confuse similar memories with each other, which is a very important memory function. Whether or not it plays a special role in Alzheimer's disease above and beyond what the other brain areas do, this is something that we are still trying to understand. There seems to be quite a intricate interplay between the functions of different hippocampal subfields, where impairment in one subfield may lead to an over-recruitment of functions in other subfields that may cause certain shifts in how you perform different memory tasks and certain biases in memory. And this is something that we are just beginning to understand. So dentate gyre is very important, interesting structure because of its potential for plasticity, because it's also reacting to interventions such as cardiovascular exercise and sport. But certainly we need to understand also its interrelationship to other to other hippocampal subfields and the entorhinal cortex in the disease context.
Wonderful. Thank you so much for all your great answers and for coming here today. You have contributed a lot to this day for us, so thank you.
Thank you very much for having me.
Da sier vi tusen takk til Emra, og vi skal videre til siste foreleser. Og nå vil jeg bare understreke helt for siste gang at her har dere altså en Alzheimers ekspert i lomma, så det gjelder å gripe både telefonen og sjansen til å faktisk stille spørsmål og stemme frem det dere vil ha til svar. Og siste foreleser, han heter Henrik Zetterberg. Han er lege og professor ved Universitetet i Göteborg. Og det Henrik skal snakke om i dag, det er det at det har foregått så store teknologiske fremskritt nå, at man faktisk har muligheten til å måle objektive forandringer på sykdom i Alzheimers tilfeller, i blodet til pasientene. Og dere kan jo da bare forestille dere hvor store implikasjoner dette kan ha for tilgjengelig og muligheter for diagnostikk. Så da sier vi bare vær så god til Henrik Zetterberg.
Thank you very much for having me as a speaker in this opening symposium. My name is Henrik Zetterberg. I'm a professor of neurochemistry at the University of Gothenburg and at University College London and the UK Dementia Research Institute. And I will talk about biomarkers for Alzheimer's disease. And with biomarkers, I mean objective measures of, in this case, brain pathology in Alzheimer's disease. And I would like to start with this slide, which shows the neuropathological changes in an Alzheimer's disease brain. And here we can start by looking at the amyloid plaques that build up in Alzheimer's disease as clumps in the brain tissue. And then we also see tangles here. Tangles are composed of hyperphosphorylated tau proteins. I'm sure you have heard a lot about tau from Karen. And then we have neurodegeneration in Alzheimer's disease also. That's basically loss of neurons. But in particular, you also lose synapses, which I think you have heard plenty of from Tara Spires-Jones earlier today also. These three brain changes are exactly what Alois Alzheimer saw in his patient, Auguste Dieter, when he examined her brain in 1906 in Germany. And this stain is actually, it's basically what he could see in his microscope. If we want to diagnose Alzheimer's disease and be very certain on the diagnosis, we have to examine the brain. But you can't sample the brain, at least not, it's not easy to do it. It's also a little bit dangerous to do it. So we would like to be able to monitor these brain changes in living people, and then we need to develop biomarkers. And that we have actually succeeded to do. So here I will start by showing you beautiful neuroimaging examinations of brains. And this is a brain where examined by something called amyloid PET. So then you have positron emission tomography, and what you do is that you inject a molecule that binds strongly to the amyloid plaques in the brain. You inject the molecule intravenously, it will pass, it will cross the blood-brain barrier and bind to the plaques. And then this molecule is labeled with radioactivity. So you can actually measure traces of this molecule in the brain. And the redder it is, the more amyloid plaques there are here. This is a tau PET imaging, so you can also tau PET scan. So you can also look at the tau tangles that you saw in the earlier slide. PET examinations are beautiful but expensive. They are not available everywhere, so they are not easy tests to use, even though they are good. We have instead been working with cerebrospinal fluid, and CSF is a clear fluid that surrounds the brain, and it's also present deep inside the brain. The neurons and other brain cells bathe in cerebrospinal fluid. This fluid is very rich in brain-derived molecules. So if we are interested in biochemical changes in the brain, we can easily, not easily, but we can in an easier manner measure those changes in cerebrospinal fluid samples instead of blood. And here you see CSF sampling. You can sample this fluid by putting a needle into the lower back of a patient. And you can do it on healthy volunteers also. It's completely harmless. One side effect is post-lumbar puncture headache, and that happens in between 2 and 10% of the subject, but it's not dangerous. In this fluid, we can measure beta amyloid, which is then the protein that builds up the senile or amyloid plaques in the brain, one of the typical changes in Alzheimer's disease brains. And then we can measure tau as a marker of neurodegeneration and phosphorylated forms of tau as a marker of tangle pathology. So basically, with these three different measures in CSF, we can detect the typical brain changes in Alzheimer's disease. And CSF and amyloid and tau PET studies have shown that amyloid plaques appear around 20 years before you're demented from Alzheimer's disease. The plaque pathology is followed by tangle pathology and neurodegeneration, and then the symptoms come. There is a window of opportunity here, because if we can detect the amyloid plaques before you have symptoms from the disease, then you could potentially detoxify them and save the brain from Alzheimer's disease. And this is exactly why we are so interested in developing simple to use blood tests for these brain changes. It will tell us a lot about Alzheimer's disease, pathogenesis, and the process from having the first signs of amyloid build-up in the brain and the full dementia syndrome, and how we could intervene before the brain is damaged in an irreparable manner by Alzheimer's disease. So we would like to have blood tests for these pathologies. Now, to be able to measure these proteins in cerebrospinal fluid, you have to have quite sensitive assays. And these assays we have had around for almost 20 years. The sensitivity of the assays is a bit hard to understand, but this is actually the best analogy. So if we take one sugar cube and put it into an Olympic swimming pool, to be able to do the CSF tests, we have to be able to measure the concentration of glucose in that Olympic swimming pool after you have added one sugar cube. If we instead want to be able to do this measurement in blood and detect the Alzheimer brain changes in blood, we will have to measure these fragments of the sugar cube added into the Olympic swimming pool. That is how sensitive our assays have to be. Fortunately, we actually have the technology to do this now. There are different technologies, but this is the one we have been working the most with. It's called single molecule array technology. This is a technique my team is working on both in Gothenburg and in London. The technique is based on antibodies, and antibodies, everyone knows about antibodies now in COVID times. Antibodies are these molecules that proteins that specifically and strongly can measure, for example, bind to viruses and bacteria and so, but we can also make them bind to certain proteins. So in this single molecular array technique, we have magnetic beads, small magnetic beads that have plenty of antibodies that are specific to our target analyte coated onto the magnetic beads. This is our target analyte. This could be, for example, beta amyloid. And then we we add the magnetic beads with the antibody to the sample, the blood sample from a patient, for example, and then we bind up the target analyte like this, and then we have another antibody to detect it. And this other antibody binds to another part of this molecule. And this antibody is labeled with an enzyme. And this enzyme can make a substrate flourish. It can give you a fluorescent product. You will get light emission. And then eventually you can detect this in microwells. And then you see here individual microwells lighting up if there is analyte present, if the molecule is present. This is detected on CDs that look like this, and the grids with the microwells look like this. So here you get a sense on what it looks like in the lab. And this is what it looks like in regards to the signal. So here you see a low amount of analyte present. Most of the microwells are dark. Individual microwells light up. And if individual microwells light up, you can actually count that there is, in most of these light emitting microwells, there is one molecule present. But you see that some actually light up twice as much, and then there are two molecules there. Here is when it's more, and here is when it's a lot. Most of the microwells have the analyte present and emit light. So by doing this, we can get a very broad dynamic range of the assay. It is very, very sensitive, and we can start to measure beta amyloid, tau, and phosphorylated tau in blood samples. So we have the brain, we have the Alzheimer brain changes, and we can now look at this in blood. This is what it looked like many years ago when we started to measure plasma beta amyloid concentrations. Beta amyloid concentration in regular blood samples. Some studies showed increased levels, most showed no change, and some showed decreased levels, but there was no clear pattern across studies. It basically meant that we, using the technologies available then, we measured noise. But in 2017, this paper came. This is a mass spectrometer-based technology where, with the use of antibodies, all beta amyloid from a blood sample from a patient was captured and then quantified with a specific mass spectrometry based technique. And then the A-beta 42/40 ratio could be seen to be reduced in the plasma of individuals with amyloid in their brains. You can see that you can actually draw a line here so that most of these individuals could be labeled as positive by the amyloid test. There are some people who are amyloid negative and have a negative test, but longitudinal studies show that they often turn out to become amyloid positive when examined with PET scans later on. So the test seems to be rather good. This is another, it's a busy slide. This is actually a Japanese-Australian. This was an American research group. This is a Japanese-Australian collaboration. They did also a mass spec-based technique where they first used antibodies to pull out all beta amyloid from the blood sample from the individuals. And then they measured the A-beta 40/42 concentration. They inverted the ratio and then they detected an increased ratio instead in amyloid positive individuals. Basically, to me, this is the same result as in the previous slide here. We have also made a test where we use standards and antibodies and add the standards to the plasma sample, and then we can immunoenrich for all of the available beta amyloids and separate them in mass spectrometry, a mass spectrometry instrument, and get a quite good test for A-beta 38, 40, and 42. These are the different forms of beta amyloids. This is the most aggregation prone. And then we did this measurement on individuals who were cognitively normal and had undergone an amyloid PET scan. They were 70 years old, and 20% were amyloid positive according to the PET examination. And with the amyloid test, we could detect most of them like this. The area under the curve, which is one measure of the diagnostic accuracy of the test, was 0.82, which means roughly that we were right in. By just analyzing the blood, we could detect 82%, classify 82% correctly in this study. So it's not a bad test. It works quite well, and it costs a fraction of what the amyloid PET scan does. Then we turned to, we also developed this test into a simple clinical chemistry test together with, this was a collaboration with Roche Diagnostics company. And the platform here is Alexius. It's present in many clinical chemistry labs in hospitals all around the globe. And you can see here that the diagnostic accuracy of the test, by adding some other parameters, you can get up to almost 0.9, not really, but between 0.8 and 0.9 in diagnostic accuracy. So it is a test that is rather accurate. And one could then think about doing this in a primary healthcare unit, and then you would be able to tell if it's very likely or unlikely that there is Alzheimer's in the brain already in a stage where there are minimal symptoms. And then at the present, one could then take positive individuals, refer them to a memory clinic, and then do CSF or amyloid PET to be certain or more certain. But this could be one way of detecting early Alzheimer's disease the day we have effective treatments. And they are around the corner. And this we can discuss more in the live session, because it's a super exciting time in Alzheimer's disease drug research just right now. So we have never been so close to potentially having the first, what we call disease-modifying treatment available. Now, phosphorylated tau, the tangle marker, can we measure that in blood? Yes, actually now we have a very sensitive and nice test for that as well. Again, it's based on the same platform I showed earlier in this talk, the Simoa assay. It's very specific for phosphorylated forms. Total tau is not detected at all. We see young individuals, they have low levels of phosphorylated tau, cognitively unimpaired elderly, they have slightly higher levels, and then we have patients with mild cognitive impairment and Alzheimer's dementia patients like this. Quite clear differences. Here is another set of individuals who donated plasma in this study. Frontotemporal dementia patients are completely normal, which is exactly what we expect for a phospho-tau test, which is really, in CSF, we knew this was AD specific. And you see the AD group is up here. So there are clear changes in the blood if you have Alzheimer's disease in the brain. And actually, to me, this is a bit of a favorite slide because Alzheimer's disease patients can sometimes be hard to differentiate clinically from frontotemporal dementia patients. But with a simple blood test, you get complete separation here, which is amazing, I think. The blood test correlates with tau PET. That is the expensive but quite nice imaging examination of tangle pathology. It correlates also with amyloid. So if you have amyloid and tau in the brain, you have a positive blood test. And in familial Alzheimer's disease, in individuals who know they will get Alzheimer's because they carry mutations that will cause Alzheimer's disease, then actually phosphorylated tau starts to increase in mutation carriers around 10 years before expected clinical onset. So here we have a window of opportunity again to detect these people and start them on treatments that will detoxify the amyloid or do something to the underlying Alzheimer's disease process, and then hopefully we could bring them down to a normal level. This is the way I really hope that this test can be used in clinical trials and eventually in clinical practice. Here we had, in this study, we had the opportunity to compare the plasma test to brain donation results 8 years later. So these were individuals who lived and they agreed to donate their brains for neuropathological examinations. They donated plasma, and we could measure plasma and then compare the plasma result 8 years before autopsy and brain examination. And then you see that this is really specific to Alzheimer's disease pathology. All these red ones have amyloid plaques and tau tangles. These ones, the green ones, have other pathologies. It's really a AD-specific test that we have developed. Now, neurodegeneration, do we have a test for that in blood? Yes, we can actually measure neurofilament light, which is an axonal protein that should be in the long extensions that neurons extend to connect to each other. Neurofilament light leaks out if there is axonal injury. And we could measure with ELISA in CSF for many years, but then with Simoa, we could get a better analytic sensitivity, and then we could start to measure low concentrations, 1 to 10 nanogram per liter here. And this is where most of us have our neurofilament light concentrations. This protein leaks out from neurons that are turned over, and it probably also is secreted to some extent. But if there is an injurious process to the brain, the levels will be higher, as I will show you now. The blood NFL levels correlate with CSF NFL levels in a nice manner like this. This is what it looks like in familial Alzheimer's disease. Here are non-carriers, pre-symptomatic carriers, and symptomatic carriers when they are in the dementia phase of Alzheimer's disease. And here you see individual cases. So here you see in red, you have mutation carriers, and you see that some of them shoot off in their neurofilament light concentrations as they approach the expected year of onset. So this is really a neurodegeneration marker that takes off like this. Black lines are non-mutation carriers. So the levels are quite stable if you do not have Alzheimer's, this type of neurodegenerative process. This test is not specific to Alzheimer's disease. It will also be increased in, for example, frontotemporal dementia and other neurodegenerative diseases, and also after stroke and brain trauma. This is what it looks like in Åpning, where we look at sporadic Alzheimer's disease. So Alzheimer's disease patients, they start off higher and they have a higher slope over time in their neurofilament light concentration. So basically, if we return to this brain pathology slide, we have amyloid plaques, we have neurodegeneration, and we have tau tangles. And now we have 3 blood tests that detect this. So you could actually now walk into a general practitioner's office and ask him or her to examine you for biomarker changes that would indicate that there is Alzheimer's in the brain. Would one recommend this now? This is also something we can discuss in the live session. And I mean, now perhaps this is of limited clinical utility, but we all hope that we will have effective treatments shortly against Alzheimer's disease. And then my hope is that these blood tests could help to get the right drug to the right patient in the general population. And Alzheimer's disease is such a common disease. So we have to have some kind of simple tests to start with. And then, of course, one could have this type of staged testing that if you're positive on these markers, you could be referred to a memory clinic and evaluated with more specific tests, and then one could discuss if there is a treatment one should start on. So all this is a little bit in the future, but it's in the very cutting edge, and I think that this center that is opening officially today will work a lot on these different types of biomarkers and perhaps also detect new tests for other neurodegenerative diseases or other pathogenic mechanisms that are active in the brain. And then we together will help advance this field and eventually make Alzheimer's a curable disease. We have to look at other neurodegenerative diseases as well, of course. And with this, I would like to thank you. In the upper part here, we have my lab in Gothenburg, and this is my London lab. And I would like to thank all of you for listening in. And I am happy to take questions in the live session now, and hope we'll have an intense discussion on what this could mean. Thank you very much.
Det veldig deilig å kunne få lov til å avslutte forelesningsrekka vi har hatt I dag med så mye fine forelesninger med en som såpass entusiastisk og optimistisk som det Henrik har vist seg nå å være. Og som dere ser så Henrik altså klar for mer. Så da går vi videre til spørsmålsrunden. Og Henrik, can you hear me?
Yes, I can.
Wonderful. Thank you so much for coming here today.
Thanks for a lovely day. I've learned a lot. It's been a great day. But you must be tired, all of you. You have been listening in now since 11.
Yeah, we're proud of all our viewers too. And so are you ready for some questions from them?
Absolutely.
Okay, then we go to the first one. Why do you spend a lot of time on biomarkers? Would it not be better to spend this time trying to find a cure? That's a kind of skeptical one for you as a start.
Yeah, that was a tough start. It is, I think, it is like this. If you think about the music industry, you have the rock stars, those are the ones developing the treatments, and then you have the roadies, those who make it possible. So I regard my team and myself as the roadies of AD drug discovery. I think the biomarkers are essential to make the drug discovery work possible. Of course, you can find a new therapeutic target in a basic science model setting. But when you want to try it out on the disease models and the patients, then you have to know what stage the patients are in and if your drug does something to the process you're studying. And then I think that these objective biomarkers will help to tell if the drug does what it should do. The biomarkers could also help to detect side effects. So that's why I think that this type of work is needed and goes hand in hand with drug development, and also biomarker work. That's sort of part of my skills. I do not know how to discover drugs, but I know how to measure protein concentrations. So hopefully we can work together and make this happen.
Wonderful. And so if effective blood tests for detecting Alzheimer's disease is readily available, shouldn't we start checking ourselves early and regularly?
Yeah, that's a great question. And the blood test would make this possible, but especially the phospho-tau tests look like they carry almost the same information as the gold standard tests to detect tau pathology in Alzheimer's disease. But the big question is what one should do with the results. And when we talk about screening, there are ethical recommendations regarding how one should do these types of tests. They should be affordable, they shouldn't cause harm, and there should be something you can do with the result. And the only thing we can do actively now in the absence of symptoms is to simply promote a healthy lifestyle, much as you mentioned. We can't change our genes. We can't choose our parents. The one way of preventing from getting Alzheimer's disease is to die young, and no one wants to do that. We want to live happy and healthy lives. And then all these regular recommendations that all of you know about, that one should be a bit physically active, one should try to maintain the general body in good health, and one shouldn't do drugs, and one shouldn't drink too much or smoke. All those recommendations are you can sort of adhere to without knowledge on your biomarker information and without knowledge on your genotype. So I think that we will need to get into a stage in this clinical world where we have a treatment that can be used to do something about the pathologies we detect. And then one could talk about early detection and potentially in the future screening, depending on how good the drugs are.
Okay, and what about when your blood tests will be implemented in the clinical routine? Do you know that? And has it been validated in naturalistic samples yet?
That's an important topic for future research. Diverse populations, all comers to a clinic. How do the tests work there? We have implemented one of the tests in clinical routine, and that's neurofilament light in plasma, and that is used by neurology clinics, for example, in patients with suspected multiple sclerosis, to monitor the effect of treatments, because with multiple sclerosis you have an axonal injury, increased plasma neurofilament light, and then you have effective treatments and you bring down the patients in their NFL level until it's normal or even a little bit lower than what is normally seen. So there the test works clinically as a test to monitor effective treatment for a neurological disease. We also see similar changes in spinal muscular atrophy, that the rare childhood neurodegenerative disease where neurofilament light levels are very high as the spinal cord degenerates because of a mutation in these kids. And then you can treat this mutation with an antisense technology that you inject in the lumbar sac. And then after a few injections, many of the children actually get a less intense neurodegenerative process, and then neurofilament light levels go down. We would love to see this in a clinical trial on Alzheimer's disease, and then we could use the test clinically for that purpose too. So neurofilament light is up and running in clinical routine. Plasma phospho-tau will be up and running during the spring, but it is not recommended for screening.
Okay. And so we have a viewer here, Tore Lund, who's wondering, using the blood tests for p-tau that you mentioned, how do you differentiate between Alzheimer's patients and patients suffering from other tauopathies? That's
a great question too. We have for a long time misinterpreted fluid biomarker levels of fluid levels of tau, actually also in our clinical practice, because we have thought that CSF tau levels reflect neurodegeneration and phospho-tau levels tangle pathology. But it has during the recent years been shown that phosphorylated tau increases in response to amyloid plaque pathology. So if you have these clumps that Tara Spires-Jones showed beautiful pictures of amyloid plaques, the surrounding neurons will react to that pathology by getting a synaptic degeneration and phosphorylated secrete tau. If you do not have the plaque pathology, you don't get the increase in extracellular fluid tau. So in other tauopathies like frontotemporal dementia or another neurodegenerative disease called progressive supranuclear palsy, which is a little bit Parkinson-like, then you have tangle pathology but no change in the tau we can measure in fluids. It's still a bit of a puzzle why it is like this, but the tau levels are Alzheimer's specific in the fluids.
And what about the potential treatments that are under development? Are there any, and which do you think have the best chance to succeed? Sorry.
Yes, there are, but it is a rocky road. It's like a roller coaster ride to follow the therapeutic field. So the one that is closest to one drug that is currently being assessed for clinical approval is called aducanumab, and that's an antibody against amyloid clumps. That drug has a very interesting story. So if you have time, you can Google on it. It has been both regarded as completely negative and very positive, and now FDA in the US is looking into if they should approve it, and EMA in Europe are also examining an application for the drug company to approve the drug. Many people were very positive a few weeks ago, but then there was an open hearing just a couple of weeks ago also, and that was very negative, and that was now I have no clue on where this will end, but that's just one of the drugs. Most of the drugs are directed against amyloid clumps. They are antibody-based, but there are also tau targeting drugs, as Karen talked about, to sort of stabilize the neuronal extensions in Alzheimer's disease. We will see where this ends, but I think we can be quite optimistic that we will have a couple of disease-modifying drugs within the next few years, and potentially there will be an American approval already before March 7, 2021. But it's super hard to predict what the final verdict will be.
Okay, so now we have time for one last question for you, and that is, do you think that cognitive tests like tests on memory or navigation could provide an added diagnostic value in combinations with the blood tests that you are talking about?
Yeah, actually, I think they are essential because these types of tests that I have described detect molecular pathologies that can occur before there are symptoms from the disease. And one extremely important thing here will be to connect the molecular change in amyloid or tau to the clinical presentation. And it's very important not to make a diagnosis in isolation on the basis of a biochemical test. You have to put the test result into a more complete picture, and cognition will be super important, and more advanced cognitive tests that will be developed in this center, that will also, together I think that will give a much clearer picture on where in the disease process an individual is. And that will also be super important to evaluate new treatments. So complete medical history, complete clinical assessment of the body and the brain, and then the biomarker tests, and then cognitive function tests, and potentially some stress tests and imaging. And such things. We must remember that these neurodegenerative diseases are serious diseases that will require quite extensive investigations, and we have to put resources on them to understand them better, to develop the drugs and more help these people as good as we can.
Da mistet jeg litt kontakten med bildet ditt, så jeg har ikke muligheten til å takke deg ansikt til ansikt, Henrik, men jeg vil i hvert fall si 1000 hjertelig takk for at du har vist all den kunnskapen du har, og vært villig til å dele den med oss og publikum i dag. Og jeg må jo også få legge til da, at det hadde absolutt ikke vært mulig å ha en så god diskusjon som det vi har hatt i dag, hvis ikke det var for de helt fantastiske spørsmålene som har vært sendt inn av dere ivrige deltakere som har faktisk fulgt oss i hele dag da. Og helt til slutt i programmet nå, så skal jeg sette over til leder for senteret vårt, Edmart Moser. Vær så god.
Jeg ønsker å takke dere alle for å ha vært med på det her. Jeg synes at vi har fått i de 4 foredragene høre ganske masse om hvorfor enkelte hjerneområder, særlig de relatert til stedsans, tidssans, hukommelse, veldig viktige i å forstå Alzheimers sykdom. Vi har forstått hvordan folk arbeider for å finne en kur mot sykdommen, og vi har forstått hvordan vi kan oppdage Alzheimer tidlig på forskjellige måter. Så med den samlede kunnskapen så synes jeg at vi har tatt et viktig første skritt mot å drive det nye KG Jebsen-senter for Alzheimers sykdom. Så da ønsker jeg å takke en hel rekke personer som har muliggjort senteret. naturligvis så vil jeg begynne med stiftelsen Christian Gerhard Jebsen. Så uten bevilgninger fra stiftelsen så hadde vi naturligvis ikke stått her i dag. Jeg var imponert over både interesse som stiftelsen viste, men også den grundige og penetrerende jobben de gjorde når de prøvde å finne ut om dette senteret ville være levedyktig. Den interessen jeg synes vedvarer, og jeg er helt sikker på at den kommer til å følge oss gjennom hele senterperioden. Jeg vil også takke styreleder Hans Peter Jebsen for hans velvalgte og veldig hyggelige ord i talen som vi hørte for litt siden. Jeg setter veldig pris på å ha den støtta i ryggen. Det er også flere som har bidratt vesentlig. For det første NTNU. NTNU stiller opp med midler og ressurser. Samme fra St. Olavs hospital og fra Helse Midt-Norge. Alle tre har bidratt finansielt til senteret og også med ressurser. Og i tillegg så har vi også fått en oppstartsbevilgning fra Nasjonalforeningen for folkehelsen, som var viktig for å komme i gang så fort som bare mulig. Så vi takker alle de også. Videre så ønsker jeg å takke de 4 foredragsholderne, Tara Spires-Jones, Karen Duff, Emra Dutzel og Henrik Zetterberg, som har gitt 4 interessante foredrag og utvidet perspektivet, og som har klart å formidle det på en folkelig måte, sånn at det er tilgjengelig for et bredt publikum. Så jeg håper mange av dere har fått utbytte av det. De 4 er også vårt rådgivende panel. De møter oss en gang i året for å gi oss råd om siste utvikling i Alzheimer-feltet, og for å diskutere vår strategi og gi oss tips til ting som kanskje kan gjøres annerledes, kanskje ikke. Så ønsker jeg også å takke de som har muliggjort det arrangementet her. Det er Rita, det er Pia, det er Karoline og det er Halvard. Uten deres innsats så hadde det heller ikke vært mulig, og det har vært spesielt krevende fordi vi begynte med å planlegge et fysisk arrangement. Det så veldig optimistisk. Vi var optimistiske i løpet av sommeren, så endret det seg. Vi planla en kombinert løsning, både digitalt og fysisk, og så ble det helt digitalt nå helt på slutten. Og hver gang vi gjorde de endringene, så måtte de 4 da hive seg rundt og planlegge på nytt. Men samtidig så har det vært en utprøving, og første gang at vi faktisk har fått produsert et slikt heldigitalt arrangement med innblikk i hvordan forskningen blir gjort, sammenvevd med de forskjellige foredragene. Så jeg håper dere også er veldig fornøyd med produktet. Og helt til slutt så ønsker jeg da å takke alle dere som har deltatt her fra den andre siden, dere som har stilt spørsmål, dere som har fulgt med. Takker veldig mye for interesse, og håper at vi møtes ved neste korsvei. Tusen takk. Ha det bra.
Kjære alle sammen, da har vi kommet til slutten av den interaktive sendingen vi skulle ha i dag. Og jeg vil gjerne benytte anledningen en gang til og få lov til å takke dere så inderlig mye for at dere har vært med på å gjøre denne dagen til nettopp det den har vært. I mine øyne så har denne dagen vært fullspekket av nysgjerrighet, informasjon og ikke minst felles forskningsglede. Og jeg er helt overbevist om at dette bare er starten på noe veldig stort og spesielt. Så helt til slutt så vil jeg bare si tusen hjertelig takk for oss.